The strike misses by the width of a fin ray. A reef shark lunges, jaws flaring, and instead of soft flesh, it meets a sudden sphere of spikes, an explosion of geometry where a streamlined body existed a heartbeat earlier. The pufferfish inhales water with violent efficiency, buccal cavity expanding, elastic stomach distending into a taut globe that doubles its apparent mass and multiplies its silhouette. What was edible becomes impossible. What was vulnerable becomes theatrical. The shark veers aside, not from fear alone, but from physics. A mouth designed for slicing cannot seal around a sphere armored with keratinous spines. For a moment, defense is absolute. And in that moment lies the seed of overconfidence. Inflation is not magic. It is biomechanics under duress. The puffer's stomach is a marvel of extensibility, its collagen fibers arranged to permit rapid distension without rupture. Specialized muscles coordinate the gulping of water, and valves prevent premature expulsion. As the body swells, spines that lay flat along the skin pivot outward, converting pliable integument into a thicket of points. Many species compound this with tetrodotoxin, a potent neurotoxin synthesized not by the fish itself, but by symbiotic bacteria housed within its tissues. The toxin binds to voltage-gated sodium channels in nerve cells, preventing the propagation of action potentials and thereby halting muscle contraction. Predators that ignore the warning can find themselves paralyzed, respiration stilled by molecular sabotage. On paper, this is a near-perfect defense, mechanical deterrence reinforced by chemical catastrophe. But evolution does not reward absolutes. It tests margins. Inflation imposes metabolic costs that are invisible in the instant of spectacle. The act demands a surge of anaerobic effort, elevating lactate in muscle and accelerating heart rate. Once distended, the pufferfish becomes hydrodynamically clumsy. Its streamlined profile, evolved for precise maneuvering among coral branches, is replaced by a sphere that drags against water with exponential resistance. Vision narrows as the eyes are repositioned by swelling tissue. Fins, now dwarfed by body volume, struggle to generate effective thrust. The fish that could once dart between crevices is transformed into a buoyant balloon, conspicuous and slow. This is the paradox of overconfidence. A defense that works most of the time shapes behavior in ways that magnify risk when it fails. On reefs where predation pressure is moderate, puffers often forage boldly, nibbling at crustaceans and mollusks in open sand patches, rather than hugging the labyrinthine safety of coral heads. Their beak-like teeth, fused plates capable of crushing shell, grant access to prey few others can exploit. That dietary advantage, coupled with reliable deterrence, encourages exposure. The fish becomes habituated to the idea that inflation will resolve conflict, and usually it does. Yet predators learn. Certain groupers and large wrasses have been observed manipulating inflated puffers with strategic bites aimed not at the armored midsection, but at the fins and eyes, areas less protected by spines and with thinner dermal layers. Some sharks have refined a tactic of flipping the puffer upside down, exploiting tonic immobility reflexes that momentarily stun the fish. Others simply wait. An inflated puffer cannot remain distended indefinitely. Maintaining that state depletes oxygen and energy. As carbon dioxide accumulates in its bloodstream, chemoreceptors signal distress. Eventually, it must deflate, expelling water and attempting to regain agility. That transition window between spherical invulnerability and regained speed is perilously narrow. The predator, patient and calculating, anticipates it. The ecological twist reveals itself here. The puffer's greatest strength creates a behavioral inertia that predators exploit not through brute force but through timing. A shark that lunges and misses does not necessarily abandon the hunt. It circles at a distance, conserving energy while the inflated fish drifts awkwardly. The reef, once a maze of escape routes, becomes irrelevant to a body too swollen to navigate it. In this suspended state, the puffer advertises its position to every opportunist within visual range. Defense has morphed into vulnerability, not because it failed, but because it succeeded too well. Tetrodotoxin deepens the illusion of invincibility. Few predators survive repeated exposure. Those that do are rare exceptions with partial resistance. The toxin's presence reduces predation attempts statistically, reinforcing the evolutionary payoff of boldness. But toxins are unevenly distributed within the fish's body. Liver and ovaries often contain higher concentrations, while muscle tissue may harbor less. Predators that evolve tolerance or selective feeding behaviors can circumvent the chemical shield. There are documented cases of certain sea snakes and tiger sharks consuming puffers without fatal consequence, suggesting either physiological resistance or behavioral precision in avoiding the most toxic organs. The myth of universal lethality fractures under selective pressure. There is also a developmental dimension. Juvenile puffers possess lower toxin loads as their symbiotic bacterial communities are still establishing. Young fish, emboldened by inherited morphology but lacking full chemical potency, may inflate with the same theatrical confidence as adults. For them, the margin of error is thinner. Natural selection acts with ruthless efficiency on these early miscalculations, sculpting caution into survivors. But caution competes with hunger. The reef's productivity fluctuates with currents and temperature. Periods of scarcity demand risk. The puffer that hesitates too often may starve before it learns restraint. Hydrodynamics adds another layer of irony. In their normal state, puffers are not particularly fast swimmers compared to pelagic predators, yet their compact bodies allow tight turning radii within reef crevices. Inflation erases that advantage. A spherical body has a high drag coefficient. It decelerates rapidly and responds sluggishly to fin input. Should a predator discover a way to circumvent spines, by biting through softer tissue at the base of fins or attacking from below where spines are less dense, the puffer has no secondary escape mechanism. There is no sprint capacity comparable to a wrasser's burst, no ink cloud like an octopus, no schooling camouflage. The defense is singular and spectacular. When it fails, there is silence. Even the act of deflation carries peril. Rapid expulsion of water must be coordinated to avoid internal injury. In some species, accidental ingestion of air during panic inflation at the surface can trap gas within the stomach, impairing buoyancy control and forcing the fish to float helplessly. In such cases, what began as a defensive reflex becomes a death sentence, as surface predators and seabirds capitalize on the stranded form. A system evolved for underwater deterrence malfunctions in the wrong medium. Context transforms advantage into liability. And yet, the story does not end with condemnation of confidence. Overconfidence is not a moral flaw. It is an emergent property of evolutionary success. The pufferfish lineage has persisted for millions of years. Its defense is effective enough to maintain reproductive continuity. Their beaks regulate invertebrate populations, preventing certain mollusks from overwhelming reef substrates. By consuming hard-shelled prey, puffers participate in nutrient cycling, breaking calcium carbonate structures into digestible fragments that re-enter ecological pathways. The very traits that embolden them contribute to reef balance. The narrative reversal lies in recognizing that defense is not merely about survival in isolation, it shapes community dynamics. Predators that adapt to exploit puffers exert selective pressure not only on the fish, but on competing prey species. A shark investing time in mastering puffer manipulation may reduce pressure on other reef fishes, altering competitive hierarchies. The puffer's inflation spectacle becomes a signal within the ecosystem, influencing predator foraging strategies and prey distribution. Its confidence ripples outward. Still, on the scale of an individual life, the margin remains unforgiving. A puffer grazing too far from shelter, confident in its ability to balloon at the first sign of threat, may encounter a predator with patience and experience. Inflation blooms, spines bristle, toxin lurks. The predator circles, unhurried. Minutes stretch as metabolic reserves dwindle. The fish, once bold, becomes exhausted. When water finally jets from its mouth and body shrinks back towards sleek vulnerability, the predator is already aligned. A single calculated bite to a less armored flank can bypass spines and deliver crushing force before inflation can recur. There is no layered redundancy, no second act. In this theater of coral and current, overconfidence is not punished every time. Most displays end in deterrence, reinforcing the strategy. That statistical success breeds behavioral boldness across generations. But evolution does not promise certainty. It tolerates risk. The pufferfish lives by a defense so dramatic that it feels definitive. Yet the ocean thrives on exceptions. A tactic that works 99 times creates a 100th scenario where its user is unprepared for deviation. In that outlier moment, spectacle collapses into vulnerability, and the reef absorbs another quiet recalibration of its balance. The inflated sphere drifting in turquoise light seems invincible, a floating fortress of spikes and poison. But beneath the surface mathematics of survival, certainty is illusion. Defense shapes behavior, behavior shapes exposure. Exposure invites innovation from those who hunt. The pufferfish's confidence is earned, yet conditional. And when the condition shifts, when a predator has learned, when energy wanes, when context changes, the brilliant shield becomes a trap of its own making. The current shifts without warning, and the anchor is gone. A seahorse that had wrapped its prehensile tail around a strand of seagrass finds the blade torn free by a surge, the plant uprooted by a pulse of water driven by distant wind and tide. For a creature built on stillness, this is catastrophe. The body tilts upright, dorsal fin vibrating in rapid, almost invisible oscillations, trying to generate thrust from a structure no larger than a fingernail. The snout points forward, eyes swiveling independently to assess threat. But the water is no longer a quiet meadow of filtered light and swaying shelter. It is an open corridor, and the seahorse is drifting. Seahorses are evolutionary contrarians. Where most fish are streamlined for speed, they are vertical, angular, armored in bony plates that resist compression but compromise hydrodynamics. Their dorsal fin beats at astonishing frequency, allowing precise hovering and micro-adjustments within dense vegetation, yet the thrust generated is minimal compared to the powerful caudal strokes of pelagic swimmers. In stable habitats, mangrove roots, coral branches, seagrass beds, this design is exquisite. Camouflage replaces velocity. Chromatophores embedded in their skin adjust pigmentation to match surrounding algae and sponges. Their bodies grow filamentous appendages that mimic plant fronds, rendering them nearly invisible to predators scanning for moving silhouettes. The strategy depends on context. Camouflage is most effective against a static background. It fails in open water where there is no pattern to merge with. When currents dislodge a seahorse from its holdfast, it enters a medium that favors speed and streamlined pursuit. Its upright posture increases drag, turning every centimeter of displacement into metabolic expenditure. The dorsal fin's rapid oscillations consume energy disproportionately relative to distance gained. Unlike schooling fish that can exploit hydrodynamic drafting, a solitary seahorse has no collective advantage. Its passivity, so effective among blades of grass, becomes exposure against the vastness of blue. Predators are attuned to anomalies. Reef fish and small sharks scan for irregular motion against uniform backgrounds. A seahorse tumbling slightly in a current creates a pattern break, an object that moves not with the smooth glide of plankton or debris, but with twitching corrective beats. The same independence of eye movement that allows the seahorse to track prey while remaining still cannot compensate for displacement. It cannot control. The nervous system, optimized for ambush feeding on tiny crustaceans, is not built for sustained escape. When threatened, seahorses may attempt to freeze. But freezing in open water is not concealment, it is suspension in visibility. Currents are not random. They are structured flows shaped by tides, temperature gradients, and underwater topography. In seagrass meadows, flow velocity is reduced by the drag of dense vegetation, creating microhabitats of relative calm. Seahorses anchor within these refuges, conserving energy and maximizing feeding efficiency as plankton drifts past. But storms, boat wakes, or seasonal shifts can alter flow regimes abruptly. A surge can uproot vegetation or bend it flat, reducing structural complexity and exposing inhabitants. The seahorse's reliance on attachment becomes a liability when the substrate itself is mobile. The ecological twist reveals that slowness is not inherently weakness, it is specialization. By moving minimally, seahorses reduce detection probability and conserve energy in nutrient-variable environments. Their tubular snouts generate suction-feeding dynamics capable of capturing copepods with astonishing precision. Rapid cranial rotation, driven by a latch-mediated spring mechanism in the neck, allows them to snap prey into their mouths faster than many fish can react. In dense cover, speed is unnecessary. Accuracy and patience suffice. The problem arises when the environment shifts faster than the organism can adapt behaviorally. Consider a juvenile seahorse recently released from a male's brood pouch. Unlike most fish, where females lay eggs and males fertilize externally, seahorse males incubate embryos within specialized pouches, regulating salinity and oxygen supply until birth. This reproductive inversion ensures high parental investment, but also means juveniles enter the world already reliant on camouflage rather than dispersal. In calm habitats, this strategy maintains population stability. But in disturbed waters, young seahorses are easily swept into channels where predators patrol. Their instinct to remain passive can betray them when mobility is required. Predatory fish such as wrasses and groupers exploit this mismatch. In seagrass beds, they probe methodically, searching for slight movements among blades. When a seahorse remains still, detection is difficult. But once displaced into open sand patches or water columns, the predator's task simplifies. A quick lunge from a streamlined body closes the gap that the seahorse cannot widen. The dorsal fin beats frantically, but acceleration is modest. The mismatch between predator and prey is stark, one built for pursuit, the other for concealment. There is also a chemical dimension. Seahorses lack potent toxins or venomous spines. Their defense is almost entirely visual and behavioral. In ecosystems where turbidity increases due to sediment runoff or human disturbance, camouflage efficacy may decline. Sediment clouds alter background coloration and light penetration, forcing seahorses to recalibrate pigmentation. During this adjustment period, contrast mismatches can occur, increasing detectability. Environmental degradation thus amplifies vulnerability by undermining the very mechanism that enables survival. Yet, once again, the narrative resists simplicity. Seahorses are not entirely passive victims of flow. Their prehensile tails are remarkably strong relative to body size, capable of withstanding significant drag forces. By anchoring to stable substrates, sponges, corals, artificial structures, they can endure currents that would displace other small fish. Some species inhabit deeper waters where flow is steadier and less turbulent, reducing displacement risk. In these zones, slowness becomes an advantage, allowing energy conservation in habitats where prey density is sporadic. The reversal crystallizes when examining predator perception. In dense vegetation, rapid movement may actually increase detection. A fish darting through seagrass leaves hydrodynamic signatures and visual trails that attract attention. The seahorse's stillness disrupts these cues. In this context, speed is liability and slowness is shield. Only when the environment transitions to open exposure does the equation invert. The same trait that ensured invisibility becomes insufficiency. Climate variability introduces additional stressors. Rising sea temperatures can alter current patterns and increase frequency of extreme weather events, dislodging habitat structures more often. As seagrass beds decline due to pollution or coastal development, the safe zones shrink. Seahorses forced into fragmented habitats encounter edges more frequently, where open water begins abruptly. At these boundaries, the demand for speed is sudden and unforgiving. The metabolic consequences of forced movement accumulate. A seahorse displaced repeatedly must expend energy to re-anchor, reducing resources available for growth or reproduction. Chronic stress elevates cortisol levels, impairing immune function. Individuals that survive repeated exposures may still suffer long-term fitness costs. The ecosystem, in turn, feels the ripple. Seahorses contribute to regulating small crustacean populations, and their decline can alter plankton dynamics in localized zones. In a quiet meadow of seagrass, the seahorse is a master of invisibility, a vertical ghost that sways with vegetation and strikes with precision. In open water, it is an artifact of misplaced design, a creature asking for time in a medium that demands speed. The mistake is not slowness itself, but failing to transition strategies when context shifts. Anchored, the seahorse thrives. Adrift, it falters. The current subsides eventually. The seahorse catches a fragment of vegetation with its tail, coiling tightly until stability returns. Chromatophores darken to match new surroundings, body angling to mimic sway. Stillness resumes its reign. For now, the environment aligns once more with specialization. But the ocean is dynamic, and safety is conditional. When the next surge comes, the question will not be whether camouflage works in theory, but whether the seahorse finds itself in cover or suspended in exposure. In this delicate balance between passivity and necessity, survival hinges on alignment between behavior and habitat. The seahorse embodies the elegance of adaptation and the fragility of specialization. Move too little in the wrong place, and the world that once concealed becomes a stage. The shell splits along a fault line no predator can see, and in that silent rupture, a fortress becomes a coffin. Beneath a ledge of cold Atlantic rock, a lobster strains against its own armor, hemolymph pressure rising as enzymes have already weakened the inner seams of its carapace. For months, it has been protected by calcified plates, hardened with calcium salts and cross-linked chitin. Now, it must tear that protection apart. Growth demands destruction, and in the minutes that follow, survival hangs on a clock that cannot be paused. Molting, or ecdysis, is not a cosmetic shedding. It is a physiological gamble. The lobster's rigid exoskeleton cannot expand, so internal tissues grow until pressure signals endocrine pathways to release molting hormones. Specialized glands initiate a cascade that reabsorbs portions of the old shell's calcium, storing it internally as gastroliths to be redeployed into the new cuticle. The animal stops feeding. It retreats, ideally into a crevice narrow enough to exclude larger predators, but spacious enough to permit wriggling free. When the dorsal carapace finally cracks, the lobster must extract not only its body, but each delicate limb, antenna, and walking leg from a hardened mold that fits like stone. In the instant of emergence, the lobster is no longer an armored crustacean, but a pliable organism encased in gelatinous skin. Its new exoskeleton is translucent and soft, incapable of resisting even moderate bite force. Muscles that once leveraged rigid joints now push against yielding tissue. Claws that could crush mollusk shells are temporarily useless, their grip imprecise without hardened articulations. The animal absorbs water to expand its body before the new shell sets, inflating itself to a slightly larger size that will define its next growth interval. This expansion increases volume but decreases control. It cannot fight. It cannot sprint. It can only hide and wait for calcification to restore its strength. The narrow survival window that follows is measured not in days alone, but in predator proximity. In reef systems and rocky substrates, fish such as cod and groupers patrol with chemical acuity, detecting subtle cues of vulnerability. The scent of a recently molted lobster, minute changes in exoskeletal chemistry and released fluids, can signal opportunity. Octopuses, with sensory arms that taste as much as they grasp, probe crevices relentlessly. A lobster that chose a refuge too shallow or too exposed may find its sanctuary transformed into a trap. Timing is therefore everything. Lobsters often molt at night, exploiting reduced visual detection by diurnal predators. Some species synchronize molting with lunar cycles or seasonal temperature shifts that influence predator activity patterns. Warmer water accelerates metabolic processes, shortening the hardening phase, but also increasing predator metabolism and activity. In colder waters, calcification may take longer, extending vulnerability, but reducing the speed of would-be hunters. The interplay between environmental temperature and exoskeletal chemistry becomes a calculus of risk. Mistime the molt in warming waters, and the shell may not harden before a predator arrives. The ecological twist reveals that molting is not merely individual risk, but ecosystem choreography. In certain habitats, clusters of lobsters may molt within overlapping periods, diluting predation pressure across multiple vulnerable individuals. Predators can consume only so many soft-bodied prey within a given window. This phenomenon resembles predator satiation seen in terrestrial insects emerging en masse. What appears as reckless synchronicity is, in fact, a statistical buffer against total loss. Yet synchronization requires environmental cues to align precisely. Disruption of those cues through climate variability or anthropogenic noise can desynchronize populations, exposing isolated individuals without the shield of numbers. Consider the structural importance of habitat. Lobsters rely on complex rocky matrices riddled with fissures and cavities. Coastal degradation that removes or smooths these substrates reduces available molting refuges. In such altered landscapes, a lobster may be forced to molt in suboptimal locations, unable to find a crevice deep enough to conceal its soft body. The mistake is not the molt itself. Growth is essential for reproduction and survival, but executing it in an environment stripped of hiding architecture. Physiologically, molting is metabolically expensive. Energy reserves must support both the hormonal cascade and the rebuilding of a mineralized shell. A lobster that delays molting excessively to avoid risk may suffer stunted growth, reducing competitive ability for mates or territory. Conversely, molting too frequently without adequate refuge increases mortality probability. Natural selection balances growth rate with survival probability, sculpting molting intervals to local predator densities and environmental stability. Predators adapt to this rhythm. Some fish have been observed frequenting known lobster shelters during peak molting seasons, exploiting predictable vulnerability windows. Octopuses exhibit remarkable spatial memory, revisiting crevices where they have previously extracted soft-shelled prey. In such interactions, intelligence and anticipation meet biochemical necessity. The lobster's endocrine system does not account for a predator's learned patrol route. It responds to internal growth signals. If those signals align with external threat, fate is sealed. There is also the danger of premature emergence. A lobster that exits its shelter too soon after molting, perhaps driven by hunger or disturbance, exposes itself before the new cuticle has fully calcified. Early movement may crack or deform the soft shell, compromising structural integrity long-term. Worse, exposure in open substrate invites attack from even smaller predators capable of penetrating soft tissue. The urge to resume normal behavior must be suppressed until the fortress is rebuilt. Impatience becomes lethal. And yet, the reversal emerges in recognizing that vulnerability is also transformation. The very process that strips the lobster of defense enables size increase, reproductive maturation, and eventual dominance within its territory. A fully hardened post-molt lobster possesses slightly greater mass and claw leverage than before. Its new shell may incorporate minerals more efficiently, optimizing strength-to-weight ratio. The cycle of fragility and fortification is the engine of its evolutionary success. In some rare instances, a molting lobster may cannibalize the discarded shell to reclaim residual nutrients, accelerating recovery. This act of recycling reinforces the closed-loop economy of marine ecosystems, where nothing is wasted. The husk that once protected becomes raw material for the future. Even in vulnerability, the lobster participates in nutrient cycling that sustains broader food webs. Yet none of this negates the razor-thin margin between growth and death. In a dark crevice beneath a granite overhang, a newly molted lobster lies motionless, hemolymph circulating as calcium salts precipitate into rigid plates. Outside, a cod hovers, lateral lines sensing microcurrents that betray subtle movement within the rock. The predator cannot breach the crevice fully, but it waits. If the lobster misjudged depth, if one antenna protrudes too far, the waiting ends in a snap. The ocean's law is indifferent to intention. Molting is inevitable. Timing is negotiable. The mistake lies in misreading the intersection of internal biology and external danger. Grow too soon in the wrong shelter during peak predator patrol, and the transformation becomes termination. Grow in perfect synchrony with concealment and environmental lull, and the lobster emerges not weaker, but renewed. The shell, once split, cannot be resealed. Commitment is absolute. In that irreversible act, the lobster entrusts survival to precision, of season, of location, of concealment. When the new armor finally hardens and the claws regain crushing force, the animal steps from shadow heavier, stronger, seemingly invulnerable once more. But memory of softness lingers in the rhythm of its life cycle. In the end, molting is not folly. It is necessity wrapped in risk. The lobster survives not by avoiding vulnerability, but by mastering its timing. The ocean does not punish growth. It punishes misalignment. And in the darkness between old shell and new, survival belongs to those who choose their moment with almost impossible accuracy. The tentacles part for a fraction of a second, and a streak of orange breaks the boundary. In that instant, the chemistry changes. Within the anemone's forest of venom-laced arms, the clownfish is untouchable, its skin coated in a precisely tuned mucus layer that masks the molecular triggers that would otherwise detonate thousands of nematocysts. Outside that living fortress, it is a small, brightly colored fish suspended in open reef water, visible against coral rubble and blue glare. The distance is barely the length of a human forearm. For a clownfish, it is the difference between sovereignty and exposure. The relationship between clownfish and sea anemone is not casual proximity. It is biochemical diplomacy. The anemone's tentacles are armed with stinging cells that fire microscopic harpoons loaded with toxins capable of immobilizing prey many times the fish's size. Most reef fish avoid them entirely. Clownfish do not. Through a process that likely involves gradual acclimation and mucus modification, they suppress the stinging response, either by lacking certain trigger proteins or by mimicking the anemone's own surface chemistry. The result is a mutualism of rare intimacy. The fish gain shelter from predators, and the anemone gains cleaning, nutrient-rich waste, and increased water circulation as the fish dart among its tentacles. But symbiosis creates boundaries. The anemone's protective zone is not infinite. It extends only as far as the reach of its tentacles. Beyond that radius, the clownfish is stripped of its shield. Its bright coloration, which may function as species recognition within the anemone, becomes a high-contrast signal in open water. The dorsal fin that flutters so comfortably within the swaying tentacles is no match for the acceleration of a wrasse or the ambush speed of a lionfish. The clownfish's body is not streamlined for long-distance sprinting. It is compact, maneuverable, optimized for darting through tentacles rather than outrunning predators. Predators understand these perimeters. Butterfly fish and groupers often patrol near anemones, aware that clownfish must occasionally venture outward to feed on plankton drifting beyond the tentacle curtain. The reef is not a static pantry. Currents shift, plankton blooms fluctuate, and hunger compels risk. A clownfish hovering just beyond the anemone's reach must constantly gauge distance, balancing caloric gain against predation probability. The safe radius is measured in body lengths, not meters. Drift too far, and the return path lengthens beyond safe reaction time. The ecological twist lies in the very strength of the symbiosis. Because clownfish rely so heavily on anemone protection, their risk assessment may be skewed by proximity. Within tentacles, they display bold behaviors, aggressive defense against intruders, even biting at divers many times their size. This confidence, reinforced by repeated survival within the anemone's embrace, can spill over into foraging behavior. A fish accustomed to safety may overestimate its margin in open water, especially when food density is high and competition intense. Hierarchy compounds the risk. Clownfish live in structured social groups within a single anemone, dominated by a breeding female, followed by a breeding male and subordinate juveniles. Access to food influences growth and potential rank ascension. A subordinate that ventures slightly farther to capture drifting zooplankton may gain nutritional advantage. The incentive to step beyond safety is not curiosity alone, but social mobility embedded in their reproductive strategy. In this species, individuals can change sex if the dominant female dies, triggering a cascade in rank. Growth rate matters. Feeding success matters. Risk becomes entangled with future opportunity. Currents add another layer of unpredictability. A surge that carries plankton outward can lure a clownfish into a brief chase, its pectoral fins beating to maintain position. But the same current can push it sideways or backward, altering the geometry of return. Unlike larger reef fish, clownfish lack the stamina for extended pursuit. Their white muscle fibers fatigue quickly, optimized for short bursts rather than sustained swimming. A minor miscalculation, one extra fin beat, can place them outside the immediate refuge window. Predators exploit this geometry of return. A grouper positioned between the fish and its anemone transforms the safe zone into a blocked doorway. The clownfish's instinct is to retreat toward tentacles, but if that path is cut, it must attempt evasive maneuvers in unfamiliar territory. The very predictability of its retreat vector becomes a liability. Predators that learn the layout of specific anemone territories can anticipate escape routes with unnerving precision. Yet the reversal emerges when we recognize that the anemone itself is not a passive shelter, but an active participant in reef dynamics. The clownfish's presence can increase anemone growth rates by delivering nutrients and deterring butterfly fish that nibble at tentacles. In return, the anemone's tentacles create microhabitats where small invertebrates thrive. The symbiosis forms a node in a larger network of energy exchange and habitat complexity. Leaving the anemone is not merely stepping out of cover, it is temporarily detaching from an ecological partnership that shapes the surrounding reef. Environmental stress complicates the picture further. Rising sea temperatures and coral bleaching events can weaken or kill anemones, shrinking the available refuge. A clownfish whose host is compromised faces a harsh choice: remain with a failing partner, or venture farther in search of a new anemone. Migration between hosts is perilous, as territory disputes with resident clownfish can be violent, and exposure time increases dramatically. The safe radius dissolves into open reef. There are moments when leaving the safe zone is not a mistake but necessity. During breeding, males must tend eggs laid on substrate adjacent to the anemone. They fan and clean them, sometimes positioning themselves partially outside tentacle coverage. The reproductive imperative demands exposure. In such cases, vigilance intensifies, and quick retreat pathways are rehearsed repeatedly. The boundary becomes a line walked daily, not crossed casually. The mistake arises when context shifts faster than instinct adapts. A juvenile chasing a drifting copepod may fail to notice a shadow passing overhead. The neural circuitry that tracks prey movement may override peripheral threat detection for a fraction of a second. That fraction is enough. The reef does not offer grace periods for delayed awareness. And yet, without stepping beyond tentacles, the clownfish would starve. Plankton density within the anemone's reach is insufficient for sustained nourishment. Growth, reproduction, and the maintenance of mucus chemistry all require caloric input sourced from outside the fortress. The symbiosis protects, but it does not feed entirely. Thus, the fish lives in a perpetual negotiation between safety and sustenance. In the choreography of reef life, the clownfish darts outward and back, tracing invisible arcs defined by predator patrol routes and current speed. Its safe radius is not static. It expands and contracts with light levels, turbidity, and the behavior of neighboring fish. At dawn and dusk, when predators shift activity patterns, the margin tightens. At midday, glare may offer concealment, but also reduce contrast detection. Every excursion recalibrates risk. Within the anemone's tentacles, the clownfish is sovereign, immune to stings that would paralyze others. Outside, it is one of many small reef fish navigating a landscape saturated with predation pressure. The symbiosis that empowers it also confines it. The safe zone is sanctuary and boundary simultaneously. A final surge of current carries plankton past the tentacle edge. The clownfish hesitates, then lunges, snapping up a morsel just beyond reach. It pivots sharply, fins beating in controlled urgency, slipping back into the waving arms of its host. The movement is precise, measured, rehearsed through countless repetitions. But precision must be constant. A single misjudged extension, a moment's overreach driven by hunger or competition, and the distance between tentacle and safety becomes a corridor of vulnerability. In this reef theater, survival is not about never leaving safety. It is about knowing exactly how far it extends. The clownfish thrives because it understands the radius of protection encoded in mucus chemistry and tentacle reach. When it forgets, even briefly, the ocean reminds it that outside the symbiotic embrace, color and confidence are not shields. The jaws open and close in slow rhythm, a pale crescent flashing between rocks where darkness should have been absolute. The Moray eel's head protrudes from a crevice that once promised concealment, its nostrils flaring, gill openings pulsing in steady respiration. To the untrained eye, it appears to be merely breathing. To everything else on the reef, it is a signal. The hiding place has become a landmark. Moray eels are ambush specialists engineered for confinement. Their elongated bodies lack pectoral fins, allowing them to thread through narrow fissures that exclude bulkier predators. Their skin is scaleless and coated in protective mucus that resists abrasion against coral and stone. Within the skull, a second set of jaws, the pharyngeal jaws, lie coiled, ready to launch forward and seize prey that ventures too close. In darkness, vision is secondary. Olfaction dominates. Tubular nostrils sample water for chemical cues, decoding the reef's invisible language of stress hormones, injured tissue, and crustacean scent trails. A properly chosen crevice is not merely shelter. It is a sensor hub, funneling currents and prey within striking distance. But shelter is only as secure as its secrecy. Reefs are maps written in coral and rock, and both predators and prey read them fluently. A moray that selects a cavity with a wide entrance or clear line of sight gains improved access to passing fish, yet sacrifices invisibility. A crevice too shallow exposes the eel's outline to hunting groupers and reef sharks that patrol habitual routes. A space too deep restricts strike radius, reducing feeding efficiency. The ideal hide is a compromise between concealment and access. When that compromise shifts, through erosion, coral breakage, or simple overuse, the refuge transforms into a predictable trap. The ecological twist emerges when we recognize that memory operates on both sides of the encounter. Preyfish learn. A wrasse that loses a conspecific to a lightning strike from a specific hole encodes that location as danger. Over time, schools alter swimming paths, giving wide berth to known moray dens. Chemical alarm cues released during predation linger, reinforcing avoidance. The crevice becomes less productive. Hunger may compel the eel to remain, trusting that patience will compensate. But patience without novelty breeds starvation. Predators too learn. Larger fish and even octopuses note the repeated presence of a moray in a particular fissure. The rhythmic opening and closing of jaws, necessary for pumping water across gills, becomes a visual beacon. What once was camouflage now signals occupancy. An octopus, capable of remarkable spatial memory, may revisit a known moray den to probe for vulnerability, inserting flexible arms into cavities to test reaction speed. If the entrance is too wide, the eel cannot defend all angles simultaneously. Its elongated body, optimized for forward lunge, is less effective against multi-directional intrusion. Currents add another dimension. Crevices positioned along predictable flow channels concentrate scent and prey, but they also broadcast the eel's own chemical signature. In stagnant water, the eel's odor may linger near the entrance, detectable by sensitive predators. In high flow zones, scent dissipates, but structural stability may be compromised by storm surges that widen openings. Environmental change can subtly degrade once perfect shelters. The moray's body is powerful but not swift in open water. Outside its crevice, it undulates with serpentine motion, capable of bursts but lacking sustained speed compared to pelagic hunters. Its defense lies in confined geometry, where its jaws can strike with explosive precision and retreat instantly into darkness. If forced into open substrate, by habitat destruction or displacement from a preferred den, it becomes conspicuous and comparatively slow. The mistake is not hiding. It is clinging to a location whose protective value has eroded. There is also interspecific complexity. Cleaner shrimp often establish stations at moray dens, removing parasites from the eel's skin and even entering its mouth without harm. This mutualistic interaction enhances the eel's health, but also advertises its location. Fish seeking cleaning services approach these stations, aware of temporary truce signals. Predators monitoring such gatherings may exploit the traffic. The Moray's den becomes a node of ecological interaction, not a secret chamber. Shelter evolves into crossroads. In heavily populated reefs, competition for prime crevices intensifies. Multiple morays may contest overlapping territories, each guarding entrances with open-mouthed displays. Dominant individuals secure optimal sites with narrow, defensible openings and deep chambers. Subordinates may be forced into inferior refuges, wider gaps or unstable rubble where risk is higher. Territorial hierarchy thus shapes survival probability through spatial allocation. The narrative reversal deepens when examining the Moray's sensory world. Unlike many fish that rely heavily on vision, Morays interpret chemical gradients with extraordinary sensitivity. They can detect minute traces of prey hidden within coral cavities, striking even when sightlines are obstructed. This reliance on smell means that a den positioned along a chemical plume can be extraordinarily effective. But if prey species alter migration routes or currents shift due to seasonal changes, the chemical richness of that location declines. The eel must either relocate or endure diminished returns. Relocation itself is perilous. Moving between dens exposes the moray to open water predators and to conspecific aggression. A larger moray may claim the vacated site, blocking return. The eel's elongated body, though flexible, is vulnerable during transit, especially if forced to cross sand patches without cover. The decision to abandon a failing shelter is as fraught as remaining within it. Habitat degradation amplifies these stakes. Coral bleaching events and physical destruction from storms or human activity can collapse reef complexity, reducing the availability of deep crevices. Morays displaced from collapsed structures may cluster in remaining cavities, increasing visibility and competition. In such altered ecosystems, once rare predation events may become frequent as hiding spots concentrate. Yet even here the moray exhibits adaptive nuance. Some species adjust activity patterns, hunting more actively at night when visual predators are less efficient. Others may utilize rubble fields rather than fixed crevices, weaving through loose stones where unpredictability compensates for structural weakness. The perception of a single fixed den as sanctuary can give way to a strategy of shifting micro-habitats. The moment of misjudgment arrives quietly. A moray settles into a familiar crevice whose entrance has widened slightly due to erosion. It senses water flow and passing fish, reassured by routine. But a shark that has traced this reef's contours over months now recognizes the pattern. The faint flicker of pale jaw, the repeated pulse at the same coordinate. Instead of passing, it angles inward, jaws aligned with the exposed head. The eel lunges, pharyngeal jaws ready, but the predator's bite force exceeds the narrow margin of retreat. Shelter is not an absolute. It is a dynamic agreement between organism and environment. A hiding place that once concealed may become conspicuous as ecological memory accumulates around it. The reef is not static stone, but a living network of interactions, each encounter layering information onto geography. The moray's survival depends not merely on finding a crevice, but on assessing its ongoing security. Is the entrance narrow enough? Is the chamber deep enough? Are currents favorable? Are predators mapping this space? These calculations are instinctive, honed by selection rather than conscious thought. But instinct can lag behind environmental change. In the shadowed labyrinth of coral and rock, concealment is currency. The moray embodies the power of ambush, the precision of sensory detection within confinement. Yet the reef watches and remembers. Prey reroute, predators adapt, currents shift. A den is safe only until it becomes known. The jaws open and close again, rhythmic and pale against stone. Around the crevice, the reef adjusts its flow of life. Somewhere beyond the edge of shadow, a predator recalculates. The difference between sanctuary and exposure lies not in the rock itself, but in whether its secrecy still holds. The claw snaps shut on empty water a fraction too late. A shadow passes overhead, and instead of burrowing or scattering, the crab raises both chelae in defiance, legs braced, carapace angled forward like a shield. Its posture is not panic, it is doctrine. For millions of years, this lineage has survived by turning confrontation into geometry. Broad frontal armor, reinforced cuticle, and crushing pincers designed to fracture shell and bone alike. In the stillness before impact, the crab commits to the oldest strategy in its evolutionary playbook: stand, threaten, strike. And sometimes that decision works, but not always. The exoskeleton is a triumph of material science. Layers of chitin embedded with calcium carbonate create a composite structure that resists compression and disperses force across curved surfaces. The carapace domes slightly, reducing stress concentration under bite pressure. Claws articulate with remarkable leverage. Closer muscles generate formidable crushing force relative to body size. Against small fish or rival crabs, this configuration is devastating. The raised posture amplifies apparent size, presenting spines and serrated edges that discourage tentative predators. It is a defensive system that has been refined through relentless selection, and in tide pools and reef flats, it often deters attack before contact occurs. But defense calibrated for one class of threat can misfire catastrophically against another. Many reef predators do not test armor head-on. Triggerfish approach obliquely, jaws specialized for prying and flipping. Octopuses apply a different calculus entirely. Their soft bodies can form around armored prey, probing for joints and membranous weak points. A crab that stands its ground exposes precisely what these hunters require: fixed orientation and predictable counterstrikes. By refusing to flee, it anchors itself in space, allowing a more flexible adversary to manipulate angles. Speed is the silent variable in this equation. Crabs are capable of astonishing lateral bursts, scuttling sideways with rapid leg coordination that can outpace certain fish over short distances. Yet the decision to fight overrides that option. Standing ground consumes cognitive bandwidth. Neural circuits prioritize claw positioning and threat display over locomotor escape. In evolutionary terms, this trade-off is sensible when armor suffices. But when facing a predator whose bite force exceeds the carapace's compressive threshold, or whose strategy bypasses the shell entirely, the commitment becomes a trap. The ecological twist emerges when we consider molting. To grow, a crab must periodically shed its exoskeleton, secreting enzymes that dissolve the inner layer before cracking free of the old shell. In the hours and days following this ecdysis, the new cuticle is soft, pliable, and catastrophically vulnerable. Calcium deposition and cross-linking have not yet restored rigidity. During this window, the crab's once reliable armor is a gelatinous membrane, and the claws, though structurally intact, lack the bracing support of hardened joints. Yet behavioral patterns do not instantly recalibrate. The neural program to stand and fight remains intact, even as the physical infrastructure that supports it has temporarily vanished. A recently molted crab that misjudges its vulnerability and raises claws in defiance invites disaster. Predators attuned to subtle cues, slightly dulled coloration, softer silhouette, may preferentially target these individuals. Fish that once hesitated now strike with confidence, their teeth penetrating tissue that would normally deflect them. The same display that previously deterred becomes an advertisement of weakness. Evolution has favored molting cycles that align with periods of reduced predation risk. Nighttime, burrow concealment, synchronized shedding within populations. But no timing is perfect. Environmental shifts, sudden disturbances, or human-induced changes in predator density can expose soft-shelled crabs at precisely the wrong moment. Armor also carries metabolic cost. Building and maintaining a calcified exoskeleton requires mineral uptake and energetic investment. In nutrient-poor environments, shell thickness may be reduced, altering defensive capacity. A crab accustomed to deterring smaller predators may find itself confronting a larger, migratory hunter whose bite force exceeds previous ecological norms. Climate-driven shifts in species distribution can introduce novel predators into established systems. Standing ground, once reliable, becomes maladaptive under new pressures. There is a further layer of irony. Fighting behavior often escalates rather than deters. A crab snapping aggressively can trigger a predatory response in fish that key on rapid motion. Instead of blending into substrate or retreating into crevices, the crab's active defense creates hydrodynamic disturbance and visual contrast. In murky estuaries where camouflage might suffice, raising claws above sediment plumes increases detectability. What appears as bravery is, in some contexts, conspicuousness. Yet the reversal cuts deeper. The crab's apparent rigidity masks a hidden adaptability. Many species possess the ability to autotomize, voluntarily detach, a limb when grasped. A claw seized by a predator can be sacrificed, muscles contracting at predetermined breakpoints to sever appendage and facilitate escape. The detached limb may continue to twitch, distracting the attacker while the crab flees. Here, the willingness to relinquish part of itself becomes a survival strategy more sophisticated than brute resistance. Standing ground is not the only doctrine embedded in its biology. Retreat through sacrifice is also encoded. However, autotomy is not without consequence. Lost claws reduce feeding efficiency and competitive dominance. Regeneration demands additional molting cycles, extending periods of vulnerability. A crab that repeatedly misjudges threats may survive immediate encounters, yet accrue long-term deficits that impair reproduction. The ecosystem records these trade-offs in population dynamics. Individuals that balance aggression with strategic withdrawal contribute more effectively to future generations. Consider also the spatial context. In complex reef structures, retreat into crevices offers protection from large predators. A crab that chooses to fight in open sand forfeits this structural advantage. Habitat fragmentation, through storm damage or human activity, can reduce available refuges, forcing crabs into more exposed confrontations. Defense strategies evolved within intricate environments falter when those environments degrade. The misjudgment is not purely individual, it is ecological. Predators exploit behavioral rigidity. Octopuses, with their distributed nervous systems and problem-solving aptitude, have been observed learning to avoid the strongest claw and instead access the softer ventral side of crabs. By enveloping prey and inserting a beak at articulating joints, they bypass armor entirely. A crab that insists on frontal defense becomes predictable prey in the hands of such intelligence. The same chelae that crush muscles cannot crush a predator that attacks from underneath. And yet, in countless encounters, standing ground prevails. A fish miscalculates and receives a crushing pinch. A smaller predator retreats, deterred by the spectacle of raised claws and impenetrable shell. Evolutionary history validates the strategy often enough to maintain it. The problem lies not in fighting itself, but in failing to recognize the moment when the equation has changed, when predator size, environmental context, or physiological state renders defense insufficient. As calcium hardens a newly formed shell over successive hours and days, the crab regains its fortress. But during that liminal phase, soft, fragile, uncertain, behavior must align with vulnerability. The individual that remains hidden, that suppresses instinct to confront, survives. The one that replays the choreography of armored dominance before its armor has cured may not. In tidal rhythms and reef shadows, the crab stands as emblem of resilience forged in mineral and muscle. Its claws are not symbols of reckless aggression, but tools honed by selective pressure. Yet even the strongest shield has thresholds. The ocean does not negotiate with pride. It measures bite force against shell thickness, speed against reaction time, vulnerability against timing. The mistake is subtle, a belief that yesterday's victory guarantees today's survival. When defense stops working, when shell softens, when predator evolves, when habitat shifts, the refusal to flee can transform bravery into fatal error. The claw rises again in the shifting light, serrated edges poised, carapace gleaming with renewed hardness. Somewhere beyond the immediate horizon of sand and coral, a predator calculates angles and force. In this perpetual contest, survival belongs not to the most aggressive nor the most armored alone, but to those who discern when to hold ground and when to vanish into the lattice of refuge. The lungs are nearly empty, and the sea is still 40 body lengths below the surface. A banded sea snake threads through coral buttresses with slow, economical undulations, its paddle-shaped tail sculling with practiced restraint, but the oxygen stored within its single elongated lung is falling toward a physiological threshold that cannot be negotiated. Every stroke burns adenosine triphosphate inside red muscle fibers adapted for endurance. Every twist of its ribless trunk compresses a respiratory reservoir that runs almost the entire length of its body, and somewhere in the silent arithmetic of metabolism, a decision is being forced. Surface now, or hunt a little longer. Sea snakes are air-breathing reptiles in a realm ruled by gills, and that simple fact imposes a metabolic constraint more unforgiving than any predator. Their lung functions not only as a buoyancy organ, but as a longitudinal air sac that stabilizes posture and stores oxygen for dives that may last tens of minutes under ideal conditions. Hemoglobin concentrations in their blood are elevated compared to many terrestrial relatives, and their tissues tolerate brief episodes of hypoxia with surprising resilience. Yet none of this grants immunity to depletion. The oxygen curve descends with quiet certainty, and the ocean does not slow its currents to accommodate a miscalculation. Unlike fish, sea snakes must puncture the surface to breathe. That ascent is predictable, and predictability is peril in open water. Large pelagic predators patrol near the air-water boundary because it is a biological bottleneck where air breathers, from turtles to marine mammals, must expose themselves. A sea snake that has extended its dive beyond optimal efficiency, perhaps lured by a hidden eel or trapped fish in a crevice, approaches the surface with muscles already fatigued. Its ascent becomes less a glide and more a compelled rise. Energy management in sea snakes is a precise choreography between foraging success and respiratory debt. Many species hunt within reef structures, probing burrows for fish and invertebrates with narrow heads that can enter fissures inaccessible to bulkier predators. This hunting strategy requires bursts of muscular effort, lateral compression against coral edges, and rapid strikes driven by venomous fangs. Venom itself is metabolically expensive to produce, composed of complex proteins synthesized in specialized glands. A prolonged hunt that yields little prey may consume more energy than it provides, pushing the snake toward oxygen debt even before its stomach is full. The sea snake's elongated lung confers remarkable dive endurance, but it also creates a buoyancy challenge. As oxygen is metabolized and carbon dioxide accumulates, internal gas composition changes subtly, altering buoyant balance. The snake compensates with tail movements and body posture, maintaining neutral buoyancy near reef faces. If fatigue sets in, that fine balance erodes. A snake that struggles to hold depth may drift upward prematurely, revealing its silhouette against the bright ceiling of the sea. The ecological twist emerges when considering the surface itself. It appears to be refuge, a thin membrane separating drowning from air. Yet the surface layer is often the most surveilled stratum in the ocean. Reef sharks and larger predatory fish exploit visual contrast against the light above, detecting dark shapes rising from below. Birds of prey, too, scan for serpentine movements breaking tension at the surface. The sea snake, compelled by its own physiology, must enter a zone where escape options narrow. Exhaustion magnifies vulnerability. Sea snakes rely on lateral undulation powered by axial muscles that run the length of their body. These muscles are rich in slow twitch fibers for sustained swimming, but even endurance fibers accumulate lactate when pushed beyond aerobic limits. If a snake extends a dive in pursuit of elusive prey, lactate levels rise, muscle efficiency declines, and coordination suffers. Upon surfacing, the act of lifting the head above water requires rapid, coordinated strokes that are less precise under fatigue. A moment's delay in submerging after inhalation can expose the body longer than is safe. Thermoregulation adds another layer of constraint. As ectothermic reptiles, sea snakes depend on ambient water temperature to regulate metabolic rate. Warmer waters accelerate metabolism, shortening optimal dive durations by increasing oxygen consumption. In tropical shallows where sunlight heats the upper layers, a snake that misjudges temperature gradients may underestimate how quickly its reserves are draining. A dive that would be safe in cooler currents becomes marginal in heated lagoons. Some sea snakes absorb a fraction of their oxygen through the skin, a supplementary mechanism that slightly extends dive time. Cutaneous respiration, however, is limited and insufficient to replace pulmonary breathing. It buys minutes, not freedom. Over-reliance on this subtle pathway may tempt a snake to prolong its descent, especially if prey density is high. But the physics of diffusion cannot outpace the demands of muscle contraction indefinitely. Predators learn patterns. In areas where sea snakes forage regularly, sharks may begin to associate certain reef structures with predictable surfacing intervals. A snake that hunts the same corridor day after day creates a temporal signature. If its dives lengthen unpredictably due to misjudged energy reserves, its resurfacing may occur in less favorable locations, open sand flats rather than complex coral where cover is sparse. There is also the risk of entanglement. Fishing lines, nets, and marine debris accumulate near the surface and within reef systems. A fatigued snake ascending rapidly may fail to detect fine monofilament lines that snag scales and restrict movement. Struggle consumes additional oxygen. Panic accelerates heart rate, compounding metabolic demand. A push beyond safe limits becomes a cascade. Yet the sea snake is not reckless by design. Its nervous system integrates chemoreception, vision, and mechanosensory input to assess environmental conditions. Many species exhibit conservative dive patterns, surfacing at regular intervals that balance risk and reward. The temptation to extend a dive often arises from opportunity. A cluster of fish trapped in a tide pool, a burrow rich with prey. Natural selection rewards boldness when it yields caloric gain, but punishes overextension when it results in exposure. The narrative reversal lies in recognizing that the greatest threat is not the predator, but the internal clock of oxygen debt. The sea snake's mastery of venom, camouflage, and aquatic locomotion cannot override the biochemical reality of aerobic metabolism. In pushing the limits of its dive capacity, it transforms the surface from sanctuary to gauntlet. A sea snake ascends through the last meters of water column, lungs compressed by depth, carbon dioxide levels stimulating the urge to breathe. It breaks the surface, inhales in a swift, practiced motion, and in that heartbeat of exposure, a shadow arcs beneath. Whether the predator strikes or veers away depends on timing measured in seconds and stamina measured in molecules of oxygen. Energy mismanagement is subtle. It begins with a longer chase, a slightly warmer current, a marginally depleted glycogen store from the previous hunt. It culminates in vulnerability at the very boundary that sustains life. The sea snake survives not by never approaching exhaustion, but by reading the invisible calculus of its own physiology before the ocean does it for him. The body slips back beneath the surface, lungs replenished, muscles clearing lactate through renewed oxygenation. The lesson is inscribed not in scars, but in restraint. In a world where every breath must be earned through ascent, the margin between mastery and miscalculation is measured in the quiet descent of oxygen saturation. The strike fails by less than the width of a fin. A frogfish explodes forward from absolute stillness, its cavernous mouth expanding with ballistic precision. Buccal cavity flaring outward in a suction event so rapid that the surrounding water collapses inward like a miniature implosion. The prey fish vanishes from sight, then reappears just beyond the closing jaws, having ridden the pressure wave without crossing the lethal threshold. In less than a heartbeat, the opportunity is gone. The frogfish settles back onto the reef, as motionless as before, but something irreversible has shifted. The energy it just spent will not be easily replaced. Frogfish are masters of stillness in ecosystems where motion defines survival. Their globose bodies are draped in skin flaps and dermal filaments that mimic sponges, algae, and encrusting corals with uncanny fidelity. And their pectoral fins are modified into limb-like appendages that allow them to walk across substrate rather than swim in open water. This is not an animal built for pursuit. It is an organism that has outsourced locomotion to deception and timing. Its elysium, a modified dorsal spine tipped with a fleshy esker, dangles before its mouth like a living lure, twitching with neurological micro-adjustments that simulate the erratic flicker of a distressed worm or shrimp. The entire predatory strategy hinges on one premise: prey will come close enough. In high-productivity waters, where currents deliver a steady flux of small fish and crustaceans, a missed strike is an inconvenience. In low-energy environments, deep reef slopes, silty lagoons, or degraded coral systems where prey density is sparse, a missed strike is metabolic hemorrhage. Frogfish possess limited endurance reserves. Their musculature is optimized for explosive contraction rather than sustained activity, composed predominantly of fast-twitch fibers that generate immense power over fractions of a second. This specialization enables them to complete a feeding strike in milliseconds, but it also means that each contraction consumes disproportionately large amounts of stored glycogen. The suction feeding mechanism of a frogfish is among the fastest vertebrate movements recorded in marine systems. When the jaws open, the volume of the oral cavity increases dramatically, creating a pressure differential that draws water and prey inward with violent acceleration. This requires coordinated cranial kinesis, hyoid depression, and rapid expansion of buccal membranes. The neural circuitry that times this cascade must integrate visual cues, hydrodynamic signals detected by the lateral line, and proprioceptive feedback from the lure's oscillation. Hesitation at any stage introduces error. Trigger too early, and the prey remains beyond reach. Trigger too late, and the prey's escape reflex, often faster than the blink of an eye, has already engaged. Low-energy environments amplify the cost of error because they restrict opportunities for compensation. Coral bleaching events and sedimentation reduce structural complexity, decreasing the number of microhabitats where small fish can congregate. In such simplified landscapes, frogfish may wait extended periods between viable prey approaches. Their metabolism, although relatively slow compared to active swimmers, still demands caloric intake. Each failed strike reduces energy reserves that must sustain prolonged inactivity. There is an inherent paradox in the frogfish's design. It appears patient, even lazy, yet its survival depends on split-second decisions executed with ruthless precision. Waiting too long to strike risks the prey detecting subtle anomalies in lure movement or body outline. Striking too late converts ambush into spectacle. Striking too early wastes energy. The predator must predict not only where prey will be, but where it will be milliseconds into the future. Repeated failure introduces a compounding effect. After a powerful strike, muscle fibers accumulate metabolic byproducts that require oxygen and time to clear. Although frogfish can ventilate their gills while stationary, recovery is not instantaneous. A second strike attempted before full recovery may be slower or less forceful, increasing the likelihood of another miss. In ecosystems where prey encounters are infrequent, such a downward spiral can become lethal. The ecological twist emerges when we consider that hesitation, often framed as caution, may sometimes be adaptive. In densely populated reefs, premature strikes could alert nearby prey to the frogfish's presence, compromising its camouflage. A single failed lunge may send a ripple of avoidance behavior through the local fish community, reducing subsequent approaches. In these contexts, waiting until prey crosses a precise threshold of proximity maximizes efficiency. What appears to be delay is actually probabilistic calculation shaped by evolutionary pressure. But that calculation assumes prey abundance. In environments where overfishing or habitat degradation has thinned fish populations, frogfish may cling to historical behavioral algorithms that no longer match ecological reality. Waiting for the perfect approach could mean forfeiting marginal opportunities that would have sufficed. Evolution fine-tunes predators to typical conditions, not catastrophic shifts. The frogfish's specialization becomes vulnerability when the statistical landscape changes. Energetically, frogfish operate on narrow margins. Unlike pelagic predators that can traverse kilometers in search of feeding grounds, frogfish are largely sedentary. Their dispersal is limited, and they often remain within confined territories for extended periods. This territoriality works in structurally complex reefs rich in prey, but constrains flexibility in impoverished systems. A frogfish that exhausts prey in its immediate vicinity cannot easily relocate without exposing itself to predation or burning precious energy. The act of waiting carries unseen costs. While motionless, frogfish rely on camouflage rather than vigilance. Larger predators, including groupers and reef sharks, may consume frogfish if detected. Their cryptic morphology reduces detection probability, but not to zero. A frogfish weakened by repeated failed strikes may lack the explosive capacity required for evasive maneuvers. Thus, hesitation that leads to starvation indirectly increases susceptibility to being hunted. There is also the question of lure fidelity. The esker must mimic prey convincingly. Subtle water flow patterns influence its movement, and frogfish adjust lure oscillations through muscular control at the base of the elysium. In stagnant or highly turbulent water, the illusion may falter. A frogfish that hesitates while attempting to compensate for suboptimal lure motion may lose the brief window when prey attention is focused. The narrative reversal unfolds in recognizing that patience is both weapon and weakness. The same stillness that defines the frogfish's predatory success becomes liability when environmental productivity declines. In thriving reefs, waiting is wisdom. In depleted systems, waiting is starvation. Consider a frogfish perched on a coral head in a lagoon where fish schools have dwindled. Hours pass between encounters. A juvenile wrasse approaches, curious about the twitching lure. The frogfish delays, calculating distance, angle, and trajectory. The wrasse hesitates too, senses something amiss, and veers away. The frogfish strikes a fraction of a second late, mouth engulfing only water. The energetic cost of that single miscalculation may represent a significant portion of its daily caloric budget. Over time, repeated failures erode glycogen stores. Body mass declines. The frogfish's once plump silhouette slackens. In severe cases, individuals may resort to cannibalism, consuming smaller conspecifics to offset scarcity. Such behavior underscores the razor-thin energetic balance governing their existence. Yet the frogfish persists because in stable ecosystems, its strategy is devastatingly effective. When prey density is adequate and timing precise, a single successful strike can yield a meal nearly equal to the frogfish's own size. Its jaws can expand to engulf prey almost as large as itself, and digestion of such a meal sustains extended fasting periods. The risk-reward ratio favors explosive ambush when executed flawlessly. The mistake is not patience itself. The mistake is misreading context, waiting for ideal conditions in a world that has shifted towards scarcity. In an ocean increasingly altered by climate change and anthropogenic pressure, the calculus of ambush may require adaptation. Whether frogfish populations can adjust their behavioral thresholds fast enough remains uncertain. The reef falls quiet again. The frogfish resumes its mimicry, blending seamlessly into sponge and coral. Stillness radiates outward like a held breath. Somewhere nearby, a small fish approaches, drawn by the subtle dance of the lure. This time, the strike must be perfect. In the economics of ambush, hesitation is debt, and debt in low-energy systems compounds toward extinction. The seagrass bed erupts before the dugong even registers the disturbance. A pressure wave moves across the shallows, subtle but decisive, displacing sand in a widening arc that ripples through the meadow of halophila and halodule. The dugong continues grazing for a fraction too long, its downturned snout ploughing methodically through sediment, uprooting rhizomes with slow, deliberate sweeps of its muscular upper lip. Its heart rate remains low, its movements economical, its entire physiology tuned to conservation rather than urgency. By the time its vestibular system processes the vibration, the water column has already changed. Dugongs are architects of patience in ecosystems built on sunlight and sediment. Their bodies are massive yet streamlined, shaped like living submarines, designed not for speed, but for endurance. A thick layer of blubber insulates them in warm, tropical seas, storing energy against unpredictable fluctuations in seagrass productivity. Their metabolism is deliberately slow, calibrated to extract maximal caloric value from fibrous marine plants that few other large vertebrates can digest efficiently. This low metabolic rate, combined with deliberate feeding behavior, allows them to graze for hours in tranquil repetition. It also slows the cascade from perception to response. Seagrass meadows are shallow by definition, rooted in sunlit substrates rarely deeper than a few meters. These habitats are productive nurseries, binding sediment, oxygenating water, and forming the foundation of intricate trophic webs. They're also acoustically transparent and visually exposed. Sound travels efficiently through shallow water, but it also reflects off the seabed and surface, creating complex reverberations that can mask directional cues. A dugong feeding with its head down relies heavily on mechanoreception along its sensitive vibrissae to detect the texture and density of grass beneath the sand. Its focus is downward, not outward. The dugong's reaction time is not slow because it is incapable of speed. When startled, it can accelerate with surprising force, its broad fluke generating powerful thrust through vertical oscillations. But this acceleration is not its default state. Its nervous system is adapted to an ecological rhythm where threats are historically infrequent and energy conservation is paramount. Sudden, novel stimuli, fast boats, large sharks, or human presence do not always trigger immediate escape responses because the statistical baseline of its evolutionary past favored calm persistence over hypervigilance. In shallow feeding areas, geometry becomes constraint. Unlike pelagic waters, where escape can occur in 3 dimensions, shallow lagoons limit vertical maneuverability. A dugong startled near the surface may have only horizontal vectors available, and horizontal escape routes may intersect with sandbars, reef edges, or coastal barriers. The same shallows that allow sunlight to nourish seagrass also compress the dugong's options. The ecological twist lies in the fact that the dugong's grazing behavior creates the very landscape that sustains it. By uprooting seagrass and leaving feeding trails, dugongs stimulate regrowth and maintain meadow diversity. Their foraging patterns prevent dominance by slower-growing species and promote nutrient cycling. In doing so, they engineer a mosaic of patches that support fish and invertebrates. But these feeding trails are also visible from above and detectable through subtle turbidity shifts. Predators can learn to associate disturbed sediment with active grazing, effectively reading the dugong's ecological signature as a map to opportunity. Large sharks occasionally patrol seagrass beds, particularly in regions where dugong populations persist. A shark does not need to outswim a dugong in open pursuit. It exploits surprise within confined space. The dugong's thick skin and size offer some protection, but a powerful ambush bite can inflict severe injury. Reaction time in such moments is measured not in muscular capacity, but in neural processing speed. How quickly the animal interprets a pressure change as threat rather than background noise. Human activity has intensified these vulnerabilities. Coastal development has increased boat traffic across shallow meadows. Dugongs, surfacing periodically to breathe, often do so with little anticipation of high-speed vessels crossing their path. Their respiratory cycle, much like that of other marine mammals, is rhythmic and deliberate. A dugong rising slowly from the seabed to inhale may not adjust trajectory rapidly enough to avoid a propeller that appears only seconds before impact. Energy economics further complicate risk assessment. Dugongs require substantial daily intake of seagrass to maintain body mass. Abandoning a feeding area prematurely due to false alarms reduces caloric gain. In ecosystems where seagrass distribution is patchy, relocating to a new meadow may involve extended travel, itself energetically costly. Thus, a dugong must balance vigilance with feeding efficiency. Excessive sensitivity to disturbance could lead to chronic energy deficit. Insufficient sensitivity could lead to fatal encounter. There is also the matter of social learning. Dugongs are often solitary or loosely aggregated, but calves remain with mothers for extended periods. Young individuals learn feeding grounds and behavioral cues from adults. If adults misjudge modern threats, such as motorized boats, calves inherit those behavioral patterns. The evolutionary timescale that shaped dugong caution did not include industrial noise or artificial hull speeds. Reaction thresholds calibrated for natural predators may be poorly suited for mechanical ones. Physiologically, dugongs possess relatively small eyes compared to body size, adapted for detecting contrast rather than fine detail in turbid coastal waters. Vision is supplemented by acute hearing and tactile sensation, but sudden high-frequency noise from engines can overwhelm auditory processing, creating confusion rather than clarity. A moment's hesitation under such sensory overload can be decisive. The narrative reversal emerges when considering that the dugong's apparent sluggishness is actually a finely tuned adaptation to a historically stable, low-predation niche. Their calm demeanor reduces unnecessary energy expenditure, allowing survival in environments where food is abundant but low in caloric density. What appears as complacency is metabolic wisdom. The problem arises when environmental volatility increases beyond ancestral norms. In a shallow bay fringed by mangroves, a dugong grazes with slow assurance. Sand clouds bloom and settle as it methodically excavates roots. A shadow approaches, not the sinuous glide of a shark, but the rigid hull of a boat casting angular distortion across the water. The dugong senses turbulence but does not categorize it as immediate peril. Its ascent to breathe intersects with the vessel's path. The margin for correction narrows to seconds. Even without human interference, natural fluctuations can convert feeding grounds into danger zones. Extreme low tides compress dugongs into narrower channels, concentrating them in predictable areas where predators can patrol efficiently. Climate-driven seagrass die-offs may force dugongs into unfamiliar territories with different predator assemblages. In such transitional zones, learned caution may lag behind new risk profiles. Yet dugongs endure because they are resilient in other ways. Their slow reproduction rate, while limiting rapid population recovery, is paired with long lifespan and strong maternal investment. Adults that survive early vulnerabilities can reproduce across decades, stabilizing populations when threats remain manageable. Their evolutionary strategy is longevity over fecundity. Still, longevity depends on recognizing risk before it closes distance. The dugong's greatest vulnerability is not physical weakness, but delayed appraisal. In shallow ecosystems where change accelerates, the cost of slow reaction increases. The seagrass meadow quiets after the disturbance passes. The dugong, uninjured this time, resumes grazing with measured calm. Its fluke stirs sediment in patient arcs, its nostrils surfacing in steady rhythm. The ocean rewards consistency, but it also punishes hesitation. In a world where shallow sanctuaries can transform into ambush corridors within moments, survival belongs to those who can sense danger before the meadow trembles. The color is a warning, but it only works in a world that still understands the language. A sea slug inches across the reef in daylight, naked in every sense that would terrify another creature. No shell, no armor, no speed. Its body pulses with electric blues and molten oranges, chromatophores blazing like neon circuitry across translucent flesh. It does not hide. It advertises. Beneath that brilliance lies chemistry stolen from prey, venom siphoned from cnidarians, toxins repurposed from sponges, chloroplasts hijacked from algae and stitched into its own tissues. In this moment, it is untouchable. But the reef is changing. Sea slugs, particularly nudibranchs, are masterpieces of specialization. Many species feed exclusively on a single type of sponge, hydroid, or soft coral. Their radulae are fine-tuned to rasp specific tissue textures, their digestive enzymes calibrated to dismantle particular chemical structures without destroying the defensive compounds they intend to recycle. In some cases, they sequester nematocysts from stinging prey, transporting these microscopic harpoons through specialized digestive diverticula to storage sacs in their serrata. There, the stolen weapons remain functional, ready to fire when the slug itself is bitten. This is biochemical piracy elevated to evolutionary art, but piracy assumes the continued presence of the host. When a sea slug evolves to feed on a single sponge species, it binds its fate to that organism's ecological stability. If warming seas bleach soft corals or ocean acidification alters sponge communities, the slug's menu collapses. It cannot pivot easily to alternate prey. Its detoxification pathways, so exquisitely tuned to one chemical profile, may fail catastrophically against another. A compound that once nourished becomes poison. The metabolic constraints are severe. Many sea slugs possess slow locomotion powered by muscular waves along a broad foot. Their pace is deliberate because their strategy is chemical dominance, not evasion. Energy budgets are modest, growth is often rapid but short-lived. They reproduce prolifically, laying ribbons of eggs that hatch into planktonic larvae drifting on currents. This life cycle disperses offspring widely, but survival hinges on landing in habitat that contains the correct prey species. A mismatch is fatal. The ecological twist lies in the fact that their apparent invincibility is borrowed. A nudibranch feeding on a toxic sponge is not inherently poisonous. It is an archivist of its environment's chemical arsenal. Remove the sponge, and the slug's defense evaporates with the next generation. Its genome encodes the machinery to store toxins, not to synthesize them independently. It is perfect for one chemical landscape and helpless in another. Some sacoglossan sea slugs take specialization even further. They practice kleptoplasty, extracting chloroplasts from algae and incorporating them into their own cells. These stolen chloroplasts continue photosynthesis for weeks within the slug's tissues, converting sunlight into supplemental energy. The slug becomes partially solar-powered, a hybrid organism straddling plant and animal physiology. It reduces feeding frequency by harvesting light. But this strategy requires stable sunlight regimes and access to specific algal species whose chloroplasts are compatible with the slug's cellular environment. Increased turbidity from sediment runoff reduces light penetration. Shifts in algal community composition disrupt the delicate compatibility between host and chloroplast. A slug that evolved to thrive in clear, shallow lagoons may starve in murkier waters even if algae are abundant, because the wrong algae cannot fuel its internal solar panels. Predators too evolve. A fish species that initially avoids brightly colored nudibranchs may, over generations, develop tolerance to certain toxins. Chemical arms races escalate. A defense that was once absolute becomes conditional. The slug cannot quickly redesign its biochemical arsenal. It is bound by the pace of mutation and selection. There is also the issue of mobility. Unlike fish or cephalopods that can migrate to new habitats when conditions deteriorate, sea slugs often have limited adult dispersal capacity. Their planktonic larvae provide distribution, but once settled, adults are largely confined to localized ranges. Rapid environmental shifts can outpace generational turnover. If a coral bleaching event eradicates the specific hydroid a slug depends on, the adults cannot travel far enough to find alternative patches before starvation. In stable ecosystems, this narrow specialization is efficient. By focusing on a single prey species, sea slugs avoid competition with generalist grazers. Their digestive systems operate with precision, maximizing nutrient extraction. Their coloration warns predators effectively within a consistent community that recognizes the signal. Specialization reduces wasted effort. The narrative reversal emerges when that efficiency becomes fragility. A sea slug that is perfectly adapted to a single sponge in a single reef zone is evolution's triumph in one context and its liability in another. The same precision that grants dominance under stable conditions becomes constraint under volatility. Consider a reef undergoing thermal stress. Elevated temperatures disrupt symbiotic algae within corals, leading to bleaching. Sponge communities shift in response to altered nutrient dynamics. The specific sponge species that a particular nudibranch relies upon declines sharply. The slug, still vividly colored, still armed with stored toxins, continues its slow patrol across coral rubble. Its defenses remain potent, but its food is gone. It attempts to feed on a related sponge species, but the chemical composition differs subtly. Detoxification enzymes fail to neutralize certain compounds. Tissue damage accumulates. The slug's bright coloration remains, but its body weakens. It is chemically armed for a war that no longer exists. Some sea slugs exhibit rapid life cycles, allowing relatively quick evolutionary responses across generations. But evolutionary speed has limits. Climate shifts and habitat degradation often occur faster than selection can operate, especially when population sizes crash due to food scarcity. Genetic diversity narrows, reducing adaptive potential. Even their reproductive strategy, prolific egg-laying, is not immune to specialization's trap. Larvae drifting in the plankton must detect chemical cues from suitable prey before settling. If those cues are absent or altered by pollution, larvae may settle in suboptimal habitats, leading to high juvenile mortality. A generation can vanish before reaching maturity. And yet, sea slugs persist because specialization also fosters extraordinary ecological roles. By consuming specific sponges or hydroids, they regulate those populations, contributing to community balance. Their presence influences competitive interactions among sessile organisms. In some systems, they prevent a single sponge species from monopolizing space, indirectly supporting coral diversity. Their chemical compounds have even drawn scientific interest for potential pharmaceutical applications, illustrating that narrow adaptations can yield broad significance. But significance does not guarantee survival. A brilliantly patterned nudibranch moves across a reef now clouded with sediment from coastal runoff. Light is dimmer. Sponge cover is patchier. A predatory fish approaches, pauses, and samples cautiously. The toxin still works. For now. The fish recoils. The slug continues. The ocean, however, is rewriting its chemical lexicon. The mistake is not being specialized. Specialization is often the pinnacle of evolutionary refinement. The mistake is assuming that the environment will remain static long enough for perfection to matter. In ecosystems increasingly defined by flux, warming currents, acidifying waters, shifting species ranges, flexibility may outrank precision. Sea slugs are living equations solved for a specific set of variables. Change the variables, and the solution fails. Their colors blaze defiantly, their stolen weapons primed, their solar cells humming quietly beneath translucent skin. They are flawless in one chapter of the ocean's story and exposed in the next. Perfection in a changing sea is a narrow bridge. The reef goes silent, and something seals itself inside a transparent shroud. As daylight drains from the water column and diurnal colors collapse into muted blues, a parrotfish slows its final circuits above the coral. Its jaws, beaks of fused dental plates, capable of scraping limestone, fall still. Its pectoral fins reduce to delicate adjustments. From specialized glands near its gills, a filament of mucus begins to spill outward, unfurling like liquid silk and expanding around its body in a glistening sheath. Within minutes, the fish is encased in a gelatinous cocoon that shimmers faintly in the moonlit current. It appears hidden. It appears insulated. It appears safe. But the ocean at night does not rely on sight. Parrotfish are architects of daylight ecosystems. By grazing relentlessly on algae that compete with coral for space, they prevent phase shifts that would otherwise tip reefs toward algal dominance. Their beaks grind through calcium carbonate skeletons, and their digestive tracts process coral rock into fine sand that settles into tropical beaches. They are diurnal engineers, metabolically active under sunlight, when photosynthetic symbionts within coral polyps convert light into energy. By night, however, that productivity ceases. Photosynthesis halts, oxygen levels in microhabitats fluctuate, and the reef's balance shifts from light-driven abundance to chemically mediated tension. The mucus cocoon is often described as a chemical shield. Secreted from goblet cells, it envelops the parrotfish in a thin, semi-permeable envelope that may mask scent and trap parasites. The prevailing hypothesis suggests that the cocoon reduces detection by nocturnal predators that hunt using chemoreception. It also creates a barrier against parasitic isopods and other invertebrates that roam after dark. In theory, invisibility is achieved not through camouflage of color, but through alteration of chemical signature. Yet invisibility is contextual. Reef predators such as Moray eels and certain sharks do not rely solely on olfactory cues. They integrate mechanosensory input from lateral lines capable of detecting minute water displacements. A sleeping parrotfish, even within a cocoon, continues to ventilate its gills. That rhythmic flow creates microcurrents. A predator attuned to hydrodynamic anomalies can interpret such signals as life beneath film. The cocoon itself is not silent. Its production requires energy, and its structure is not rigid. In strong currents, it flutters slightly, refracting ambient water movement in patterns that may betray its presence. A gelatinous envelope suspended among coral branches can appear as a discontinuity in otherwise laminar flow. For a predator navigating by feel rather than sight, such discontinuities are readable. The ecological twist lies in the assumption that chemical masking equals immunity. Mucus may dilute or obscure scent, but it cannot erase metabolic heat, electrical fields, or vibration entirely. Some predators possess ampullae of Lorenzini, electroreceptive organs sensitive to bioelectric fields generated by muscle activity. A resting parrotfish produces a weaker field than an active one, but it is not zero. The cocoon does not insulate electricity. Night on the reef is a temporal inversion of power. Diurnal grazers become stationary. Nocturnal hunters emerge. Many parrotfish wedge themselves into crevices before secreting their cocoons, reducing exposure from multiple angles. However, competition for secure recesses can force individuals into suboptimal locations. Outer coral heads, exposed slopes, or sandy flats, where anchoring options are limited. In such spaces, a cocoon may function more as declaration than disguise. Energy economics also play a role. Producing mucus incurs metabolic cost. In nutrient-rich systems, this cost is marginal. In degraded reefs where food availability fluctuates due to coral bleaching or overfishing, the nightly investment may strain reserves. A parrotfish weakened by daytime scarcity may secrete thinner or less consistent cocoons, reducing effectiveness. Repeated stressors compound risk. Predator learning complicates the picture. Moray eels, for instance, patrol habitual sleeping sites, memorizing the spatial layout of crevices where parrotfish cluster. Even if chemical masking is partially effective, spatial predictability undermines it. A predator that knows where prey sleeps does not require strong scent cues. It requires only persistence. There is also the phenomenon of parasitic exploitation. Certain nocturnal crustaceans have been observed penetrating mucus layers to access host tissue. While the cocoon deters many external parasites, it is not impenetrable. The boundary between shield and liability is thin. The narrative reversal emerges when considering that the cocoon may function less as fortress and more as compromise. Parrotfish cannot remain active at night because their vision and feeding apparatus are optimized for daylight grazing. Remaining awake and mobile would increase energy expenditure without caloric return. Sleep conserves resources. The cocoon represents a strategic allocation of limited energy toward probabilistic risk reduction, not absolute safety. A reef at midnight pulses with unseen currents. A reef shark glides along the contour of coral, its lateral line mapping turbulence like topography. It senses a faint irregularity, an oscillation where none should exist. It pivots, approaching a branch of Acropora where a parrotfish rests encased in mucus. The shark's snout brushes the cocoon, and the fragile envelope ruptures in a silent burst. The parrotfish awakens in shock, muscle fibers contracting an explosive escape response. If the crevice behind it is deep enough, it may evade capture. If not, invisibility dissolves into consequence. Parrotfish survival strategies extend beyond cocoons. Their scales reflect moonlight in muted tones, and their bodies often match the substrate color. Some species change coloration at night, adopting subdued hues that reduce visual contrast. But night vision among reef predators is acute, and contrast detection under low light is efficient. Climate change introduces additional variables. Warmer waters alter metabolic rates, potentially increasing nighttime oxygen demand. Acidification can affect coral structure, reducing the availability of secure sleeping crevices. As reefs degrade, parrotfish may be forced into more exposed resting positions, increasing reliance on cocoons that were evolved for structurally complex habitats. Despite these vulnerabilities, parrotfish remain indispensable to reef resilience. By grazing algae, they facilitate coral recruitment and maintain biodiversity. Their daytime labor supports the very structures that provide nighttime refuge. This interdependence underscores a delicate feedback loop. Healthy reefs offer better hiding spaces, and active parrotfish maintain healthy reefs. The mistake is not secreting a cocoon. It is assuming that concealment eliminates risk. The ocean at night is an arena where sensory modalities overlap and predators exploit redundancy. Chemical masking may deceive some hunters, but not all. Hydrodynamic signatures persist. Electrical fields hum faintly. Spatial memory endures. As dawn approaches, the cocoon dissolves in gentle currents. The parrotfish emerges, jaws ready to resume scraping limestone into sand, resuming its role as diurnal engineer. It survived another night, not because it was invisible, but because probability favored it in a complex web of sensory interactions. The reef brightens, and with it returns the illusion of security. But every sunset will demand the same wager, that a thin sheath of mucus and a quiet crevice are enough in a world where invisibility is always partial. The shadow glides like a continent drifting through water, wings spanning wider than a small boat, cephalic lobes unfurled to funnel plankton into a cavernous mouth that never seems to close. Everything about the manta ray suggests invulnerability. It is vast, slow in appearance, immune to the twitching anxiety of smaller fish that dart and scatter at every flicker of threat. For a moment, it looks untouchable. Then the current narrows. The reef wall rises. The open blue contracts into a corridor. And suddenly, size is no longer armor. It is geometry. Manta rays are built for distance, not confinement. Their enormous pectoral fins generate lift in a manner more akin to avian flight than fish propulsion, transforming forward motion into gliding efficiency across expansive pelagic highways. Their cartilaginous skeleton is lightweight yet strong, optimized to support a body that can span several meters while remaining agile in open water. Ram ventilation forces water continuously over their gills as they swim, extracting oxygen even at modest speeds. In wide oceanic spaces, this design is elegant and efficient. In narrow channels or reef passes, it becomes restrictive. The assumption of safety begins with scale. Few reef predators can challenge a fully grown manta in open ocean. Their dermal denticles provide some protection. Their mass deters casual attack, and their sweeping fins can accelerate them away from moderate threats. But predators do not rely solely on frontal confrontation. Large sharks, including species capable of targeting sizable prey, understand spatial constraint. In open water, a manta can bank, dive, and rise with sweeping arcs that use its wings to exploit vertical gradients. In confined environments, canyons, channels, or near the seabed, those arcs shrink. Turning radius increases relative to available space. Lift requires room. Hydrodynamics imposes law. A body with wide lateral span experiences significant drag when attempting sharp turns in tight corridors. The manta's pectoral fins, though powerful, cannot fold compactly like a fish's fins to thread through narrow rock formations. In strong currents, a manta entering a constricted passage may find itself compressed between rock faces, its glide path limited to forward motion. Predators position themselves at choke points where maneuverability declines. The mistake is not entering the channel. It is misreading how geometry alters vulnerability. There is also a behavioral component. Manta rays often visit cleaning stations on reefs where smaller fish remove parasites from their skin and gill slits. These stations are social hubs, frequented by multiple individuals that circle slowly above specific coral heads. During cleaning, mantas reduce speed and alter posture to allow access to their surfaces. Attention shifts inward, focus on comfort rather than vigilance. Sharks and other large predators may patrol these predictable gatherings, aware that concentration and repetition create opportunity. The manta's trust in familiar cleaning sites can erode the unpredictability that typically protects it. The ecological twist reveals that apparent slowness is strategic efficiency. Mantas are filter feeders, consuming vast quantities of microscopic plankton by cruising through dense aggregations. Their cephalic lobes guide food into the mouth with remarkable precision, and their gill rakers trap prey while expelling excess water. This feeding strategy demands continuous forward motion and broad sweeps through water columns rich in nutrients. In such contexts, speed is unnecessary. Endurance and glide are paramount, The open ocean is their refuge because it complements their design. Yet the ocean is not uniformly open. Coastal upwellings, reef passes, and seamounts create nutrient-rich zones that attract plankton and therefore mantas. These same zones attract predators. The vertical relief of underwater mountains funnels currents, concentrating life into narrower bands. A manta entering these feeding grounds must navigate terrain that was not the original blueprint for its body plan. In deep water, vertical escape is always an option. In shallow passes, the seabed rises sharply, removing that vector. There is an additional physiological constraint. Mantas cannot remain motionless for long periods because they rely on ram ventilation. If a predator forces a manta into abrupt directional changes that disrupt forward flow, oxygen extraction efficiency declines. Unlike many sharks, mantas lack the capacity to pump water over gills while stationary. Sustained evasion in confined space risks hypoxia. Thus, a predator that pressures a manta in tight quarters does not need to overpower it immediately. It can exploit fatigue. Juvenile mantas face amplified risk. Smaller body size offers less deterrence while still limiting agility relative to reef fish. Their learning curve includes recognizing which passages are safe and which are ambush corridors. Individuals that misjudge may survive encounters but carry scars, evidence of bites that did not kill but signaled miscalculation. Those scars record the geometry of survival. There are moments when size does protect. In open ocean, a large manta's mass discourages most attackers. Its wings generate impressive bursts of acceleration when needed, and its dorsal surface blends into the dimness from below, while its pale ventral side diffuses light from above. Camouflage complements scale. But camouflage fails when background contrast sharpens against reef walls. In shallow waters, sunlight filtering through narrow gaps outlines the ray's shape distinctly. Its silhouette becomes readable. The reversal crystallizes when considering cooperative dynamics. Mantas often travel in loose aggregations. In open water, this creates a diffuse network of sensory awareness, individuals adjusting to subtle cues from neighbors. In confined spaces, however, aggregation can increase congestion. Multiple large bodies navigating a narrow channel reduce escape options. A predator targeting the rear individual in a line may exploit the limited turning space of those ahead. The collective that enhances safety in one environment can amplify vulnerability in another. Environmental change further complicates perception of safety. Coastal development and increased boat traffic can alter traditional feeding grounds, compressing manta movements into smaller areas. Noise pollution may interfere with subtle sensory cues that inform orientation. Artificial structures can create novel corridors that appear similar to natural passes, but lack predictable current patterns. An animal evolved for vastness may encounter edges it does not instinctively recognize as hazardous. And yet, the manta is not defenseless. When threatened in open water, it can perform sudden barrel rolls and steep dives that disrupt predator trajectories. Some individuals have been observed leaping from the water, possibly to dislodge parasites or avoid immediate threat. Their intelligence, demonstrated through problem-solving and complex social interactions, suggests capacity for learning from encounters. A manta that survives an ambush may alter future route selection. The danger lies in misreading scale as immunity. A body that dwarfs reef fish may still be outmatched by a great white shark or a tiger shark in the right context. The assumption that grandeur equals protection can lead to complacency in route choice. The ocean respects no silhouette. In a narrow channel between volcanic outcrops, plankton-rich water pulses with life. A manta enters, cephalic lobes unfurled, feeding rhythm steady. The walls constrict gradually. A shark positions itself at the exit, leveraging the channel's bottleneck. The manta senses disturbance, wings beating harder, but turning space is limited. It attempts to bank, but the rock face denies full arc. For a moment, mass feels like anchor rather than shield. Open water is freedom. Constriction is calculation. The manta's survival depends on recognizing when the ocean is wide enough to accommodate its grace. When the sea narrows, elegance must give way to caution. Size alone does not confer invulnerability. It dictates the dimensions of escape. The ray emerges from the channel back into blue expanse, wings resuming their effortless glide. The illusion of invincibility returns with the horizon, but the memory of constriction lingers in the rhythm of its movement. Safety is not a function of magnitude, it is a function of space. The formation fractures by a single body length and the ocean notices immediately. A silver flank drifts half a meter off alignment, a hesitation in rhythm no longer synchronized with the tight vortex of muscle and instinct that is the school. For a heartbeat, nothing happens. Then the water tightens. A dark silhouette accelerates from below, not toward the center of the swirling mass, but toward the anomaly at its edge. In the pelagic world, predators do not attack the group. They attack the mistake. Tuna are engines wrapped in skin. Their bodies are shaped like ballistic instruments, fusiform, tapered, built for sustained velocity across vast open expanses where there is no reef, no vegetation, no rock to hide behind. Unlike many fish, they maintain a degree of regional endothermy, using countercurrent heat exchange systems to warm their red swimming muscles above ambient water temperature. This physiological innovation allows prolonged high-speed pursuit, extending endurance in cold waters where competitors fatigue. But speed alone does not guarantee safety. It must be synchronized. The school is not chaos, it is hydrodynamic architecture. Individuals position themselves within a lattice of vortices created by neighbors, exploiting reduced drag and increased thrust efficiency. Each fish adjusts its tail beat frequency based on lateral line input, maintaining spacing with exquisite precision. Vision integrates flashing reflections from adjacent scales, producing a coordinated shimmer that confuses depth perception for predators. Within this kinetic sphere, survival probability rises dramatically. A predator striking into the dense center faces a wall of muscle that shifts like liquid metal, each individual buffered by collective momentum. The advantage collapses the instant formation breaks. Predators such as sharks, billfish, and dolphins do not waste energy charging blindly into cohesive schools. They circle. They probe. They create micro-disturbances, short lunges or sudden directional shifts that test the integrity of the formation. The goal is not immediate capture, but fracture. When a tuna hesitates, perhaps adjusting to a minor injury or reacting a fraction too slowly to a directional cue, the spacing widens imperceptibly. That widening is a signal flare. The predator's brain, wired for edge detection and motion contrast, locks onto deviation rather than mass. The individual that drifts becomes a solitary target in an ocean of synchronization. The ecological twist is that the school itself generates pressure. Maintaining position demands relentless attention and metabolic investment. Tuna swim continuously. Their gill ventilation depends on forward motion, forcing them to keep moving even at rest. This obligate ram ventilation creates a physiological baseline of exertion. Within the school, individuals must match velocity precisely or risk collision and displacement. Fatigue, even momentary, can degrade alignment. A fish that reduces tail beat amplitude for a few seconds may slip to the periphery. In high-density predator zones, such micro-fatigue events become lethal. There is also a cognitive dimension. Tuna process rapid streams of sensory information, visual flashes, water pressure shifts, chemical cues, all while sustaining speed. Neural processing under constant motion demands energy. In chaotic predator encounters, split-second decisions determine whether to turn inward or outward. Turning inward reinforces cohesion. Turning outward may seem like escape, but outward vectors are predictable. Predators anticipate that prey at the edge will flee laterally or away from perceived threat, not toward the dense interior. Thus, the act of breaking formation aligns precisely with the hunter's intercept model. The cost of separation is not linear, it is exponential. Within the school, predation risk per individual diminishes through dilution effect. The statistical likelihood that any one fish is targeted decreases as group size increases. Breakaway and dilution evaporates. The separated tuna becomes 100% of the predator's focus. Speed that once blended into a blur becomes a singular trajectory to calculate and intercept. Tuna are capable of astonishing acceleration, their crescent-shaped tails generating thrust through powerful oscillations of red muscle fibers. Yet acceleration outside the school lacks buffering turbulence. Hydrodynamic noise increases. The predator, attuned to pressure waves, reads the lone fish's wake like a signature. A shark does not need to outrun the entire school. It needs only to anticipate the path of the isolated individual. In open water, there is no cover, only velocity and vector. Predators exploit not only physical separation, but psychological fragmentation. Dolphins hunting tuna often employ coordinated herding strategies, compressing schools into tighter balls before triggering panic bursts. In the ensuing chaos, individuals may bolt outward, seeking escape beyond the pressure. Those bursts are precisely what predators await. The school's survival depends on resisting instinct to scatter. Cohesion under pressure is defense. Fragmentation is invitation. Yet formation is not static perfection. Environmental variables, temperature gradients, oxygen concentration layers, and prey distribution shape school dynamics. Tuna often pursue smaller fish and squid, and feeding frenzies can disrupt alignment as individuals lunge at prey. In moments of abundance, hunger overrides discipline. The very act of hunting can unravel defensive geometry. Predators shadow these feeding events, aware that focus on prey increases vulnerability to becoming prey. The narrative reversal deepens when we consider that the school is not purely defensive, but also an energy strategy. By swimming in formation, tuna reduce individual drag and conserve energy over long migrations. This efficiency supports their vast range across ocean basins, allowing them to exploit distant feeding grounds. Break formation too often, and cumulative energy expenditure increases, reducing growth and reproductive output. Thus, the pressure to remain within the group is both immediate and evolutionary. The mistake of separation carries not only predation risk, but long-term fitness cost. There are rare moments when breaking formation is advantageous. A tuna pursued relentlessly may dive sharply into cooler, oxygen-rich depths where larger predators struggle to maintain speed due to thermal constraints. Some species exhibit vertical migrations that exploit predator blind zones near thermoclines, where light refracts unpredictably. In these cases, departure from the school is strategic, not accidental. But such maneuvers require precise timing and environmental awareness. Random drift is not strategy. It is error. Human fishing practices have illuminated this vulnerability with ruthless clarity. Purse-seine nets target entire schools, yet longline fisheries and opportunistic predators capitalize on stragglers. Even within fishing fleets, captains know that the edges of a school are where the catch begins. The pattern is universal. Predators and hunters focus on the periphery. Back in the open ocean, the separated tuna feels the shift before impact, the subtle pressure wave of a predator accelerating behind it. It attempts to correct, tail snapping into higher frequency. But the school has already veered away, cohesion intact without the stray. Rejoining requires crossing open water exposed. Every meter traveled alone magnifies risk. The ocean is indifferent to individuality. It rewards synchronization, punishes deviation. The tuna's brilliance lies not merely in speed, but in disciplined alignment. Within the school, a fish is a pixel in a living storm cloud, its outline blurred by collective motion. Alone, it is a shape with edges. And yet formation demands trust. Each individual must commit to the choreography, surrendering independent trajectory to group logic. In evolutionary terms, this is a profound compromise. Survival through conformity. The moment ego, whether driven by hunger, fatigue, or miscalculation, nudges a fish outward, conformity dissolves, and so does safety. The predator closes the gap in a calculated arc, jaws aligning with the lone silhouette. The rest of the school tightens reflexively, their synchronized turn creating a ripple of silver light that dazzles and deflects. They survive because one did not. It is a brutal arithmetic embedded in pelagic life. Speed without formation is merely motion. Formation without speed is stagnation. Together, they create resilience. The mistake is not swimming slower or faster, it is losing alignment at the wrong moment. In the vast blue expanse where there is nowhere to hide, survival belongs to those who remain indistinguishable from the storm of muscle around them. The surface detonates without warning. A bloom of white spray erupts where calm blue existed a heartbeat earlier, and in that fraction of a second between inhalation and impact, the arithmetic of survival collapses. The seal had done what every mammal must do: rise for air. Yet in the ocean, breath is not merely oxygen drawn into lungs, it is a signal flare, a declaration of position, a predictable punctuation mark in a pattern that predators have memorized with ruthless precision. To live in water with air-breathing lungs is to accept a biological constraint that cannot be negotiated. The seal's bloodstream can tolerate carbon dioxide for only so long before chemoreceptors in its brainstem ignite an irresistible command to surface. That command is both salvation and sentence. Beneath the water, the seal is engineered for stealth and compression. Its blood carries an extraordinary reservoir of oxygen bound to hemoglobin and myoglobin, allowing dives that can stretch beyond what most terrestrial mammals could endure without cellular damage. Peripheral vasoconstriction shunts blood toward the brain and heart, slowing metabolism in muscle tissues to conserve energy. The lungs collapse under pressure at depth, reducing nitrogen absorption and protecting against decompression sickness. It is a physiological ballet refined by millions of years of marine life. And yet, for all this sophistication, the seal must eventually return to the one place where its body becomes conspicuous: the thin membrane between ocean and sky. Predators understand this constraint with chilling clarity. The great white shark does not patrol randomly. It patrols along vectors of probability. It studies bathymetry, current, and light. It learns that seals frequent specific haul-out rocks and feeding grounds, that they prefer certain channels where fish concentrate, that they surface in intervals dictated by dive duration and foraging intensity. The shark's electroreceptors detect faint bioelectric signatures long before vision resolves a silhouette. Its lateral line senses turbulence from a distant kick, and so the predator positions itself below, where light fractures into glare and contrast vanishes. It waits in the architecture of the abyss, calculating not the seal's strength, but its schedule. The fatal mistake is rarely dramatic in origin. It is microscopic in timing. A seal that surfaces a fraction of a second later than usual, lungs burning just enough to alter ascent speed, creates a different angle of emergence. Instead of breaking the surface at a steep vertical burst, minimizing exposure, it arcs forward slightly, presenting a broader cross-section of its body. That subtle geometry transforms it from a fleeting blur into a defined target. Hydrodynamics become destiny. The shark, built like a torpedo of cartilage and muscle, accelerates from depth with explosive anaerobic power, its red muscle fibers fueling a short, violent sprint. The strike is not blind fury. It is physics meeting biology at the exact coordinate where routine has betrayed its owner. There is another cruelty at play: surface blindness. From below, the world above is a mirror. Sunlight reflects and refracts, creating a blinding dome that conceals what lies beneath. From above, the seal scanning for danger faces glare and glittering distraction. The retina, adapted for dim underwater conditions, struggles against the harsh contrast of reflected sun. The predator below is cloaked not by camouflage alone, but by optical law. The seal looks upward for birds, sideways for conspecifics, perhaps backward for competitors, but downward, into the wash of white light and distorted shadow. Vision fails. The ocean weaponizes physics. The very act of seeking safety at the surface diminishes the seal's capacity to perceive what hunts it. Yet here lies the narrative reversal. The seal is not merely a victim of inevitability. Its predictability is a liability, but its unpredictability is a defense. In colonies where predation pressure is intense, seals alter surfacing patterns in subtle, statistically meaningful ways. They vary dive durations, introduce erratic zigzag ascents, and breach at unexpected distances from haul-out sites. These behavioral adaptations are not conscious strategies in the human sense, but emergent responses shaped by selection. Individuals that break routine survive more often, and their genes propagate. Over time, the predator's calculation becomes more complex, its model of probability less certain. The seal's weakness, its need for air, becomes the crucible in which innovation is forged. Still, innovation has metabolic costs. Erratic diving consumes more energy. Longer submergence increases carbon dioxide accumulation and lactic acid buildup, demanding longer recovery intervals at the surface. The seal must balance oxygen debt against predation risk. Too cautious, and it starves. Too bold, and it is torn in half. The ocean enforces this balance with merciless consistency. Foraging success requires repetitive routes to fish schools that move with tides and temperature gradients. The seal's whiskers, exquisitely sensitive vibrissae, detect minute water movements left by passing prey. Its hippocampus maps underwater terrain in cognitive detail. Routine enhances efficiency, but routine also sketches a map that predators can read. In regions where killer whales patrol, the calculus shifts again. Orcas do not rely solely on ambush from below, they coordinate. A pod may herd seals away from ice flows, generating waves to wash them into open water. Here, surfacing is not the only predictable act. Resting locations and escape platforms become focal points of attack. The seal, aware at some level of acoustic cues, low-frequency calls traveling vast distances, must interpret a sonic landscape filled with threat. Its auditory cortex processes underwater sound with precision. Yet even this acuity cannot always distinguish distant curiosity from imminent assault. When an orca breaches in pursuit, the seal's sprint to land becomes a desperate race against hydrodynamic superiority. The ocean is no longer a neutral medium. It is an ally of the hunter. But the seal's vulnerability has shaped the entire ecosystem. Shark predation near seal colonies influences where and how seals feed, which in turn affects fish distribution and trophic cascades that ripple outward. When seals avoid certain bays during peak predation hours, fish populations may surge locally, altering plankton dynamics and nutrient cycling. The simple act of surfacing, of choosing when and where to breathe, carries consequences far beyond individual survival. A mistimed ascent does not only end a life, it reinforces selective pressures that sculpt community structure. The ocean punishes routine because routine is legible. In evolutionary terms, legibility invites exploitation. The seal's physiology demands rhythm: dive, surface, inhale, repeat. The predator thrives on pattern recognition. Each successful ambush refines its understanding of timing, depth, and angle. Over years, perhaps decades, a seasoned shark develops a hunting strategy so precise that it appears almost preternatural. Yet even here, reversal lurks. A shark that specializes too narrowly may struggle if seal behavior shifts due to environmental change. Warmer waters alter fish migration, forcing seals into new territories. Human disturbance changes haul-out availability. Suddenly, the predator's memorized map becomes obsolete. What once was predictable dissolves into uncertainty. For the seal, the moment of surfacing remains the razor's edge. Its nostrils open in a reflexive flare, lungs expand, oxygen floods into blood starved by anaerobic metabolism. The sensation is relief incarnate, and in that relief lies distraction. The neural reward of breath can dull vigilance for a split second. It is enough. A shark striking from below can breach fully from the water, jaws agape, transforming sea into sky in a violent inversion. Gravity reasserts itself as predator and prey crash back into the depths, blood dispersing in crimson ribbons that signal to scavengers the completion of a transaction as old as marine mammals themselves. Yet not every breach ends in death. Many seals survive attempted strikes, bearing crescent scars that testify to near misses. Their subcutaneous fat layers absorb some impact. Their flexible spines allow contortions that deflect teeth. Survival imprints caution into neural circuits. Young seals learn from observing adults' timing and vigilance, imprinting social knowledge that complements instinct. The colony becomes an information network, where alarm behaviors and spatial preferences encode collective memory of danger zones. In this way, the fatal mistake of one individual becomes the lesson of many. The ocean's lesson is uncompromising. Necessity creates exposure. To breathe is to reveal oneself. To repeat is to be predicted. The seal's existence is a negotiation between metabolic law and predatory intelligence. A breath taken slightly off angle, in the wrong patch of glare, at a time when shadows align beneath. These infinitesimal variables determine whether lungs fill with air or water tinged with blood. The sea does not forgive arithmetic errors. It magnifies them. And yet, in that relentless calculus, life persists. Seals continue to surface, to inhale, to dive again into the shifting blue. Their survival is not perfection, but adaptation, constant, subtle recalibration against a world that watches and waits. Every ascent is a gamble measured in heartbeats and photons. Every breath is both triumph and risk. The ocean keeps score in silence, and the line between ordinary routine and final mistake is thinner than the surface film that separates sky from abyss. The cliff edge collapses beneath 100 bodies at once. Black and white shapes launch in synchronized arcs, flippers pressed tight, torsos streamlined, gravity converting hesitation into momentum. For a heartbeat, they hang suspended between two worlds, awkward land and lethal sea, before striking the surface in explosive bursts of foam. Beneath that foam, shadows are already moving. The ocean does not wait for hesitation. For a penguin, the transition from colony to current is not merely a commute. It is an ecological threshold where survival probabilities pivot sharply. On land, their upright posture and dense plumage insulate them from polar wind and terrestrial predators, but their biomechanics are tuned for water, not rock. In the sea, they transform into hydrodynamic projectiles, capable of extraordinary acceleration driven by powerful pectoral muscles that evolved from flight apparatus into aquatic propulsion systems. Yet that transformation requires commitment. The instant of entry is irreversible. Penguins operate at the interface of two vastly different sensory regimes. On land, visual acuity is optimized for contrast against snow and rock, and auditory cues carry across open colonies. In water, light refracts, sound travels faster and farther, and three-dimensional movement defines escape. The act of diving compresses these perceptual adjustments into fractions of a second. A penguin that leaps without sufficient reconnaissance enters an environment where predators exploit the physics of surprise. Leopard seals patrol beneath ice edges and near colony outflows precisely because they understand predictability. Penguins gather at the shoreline in tight groups, often hesitating at the lip of the water in a behavior that appears almost comical. Shuffling, craning necks, waiting for another to jump first. This delay is not indecision, but risk assessment. Individuals scan for surface ripples, for the angular distortion of a submerged predator's body against the light gradient. But once the first penguin commits, social facilitation triggers a cascade. The safety of numbers is both shield and trap. Hydrodynamics during entry create temporary vulnerability. When a penguin pierces the surface, it generates turbulence and bubbles that momentarily obscure its own vision. Its eyes must transition from air-adapted focus to underwater acuity, adjusting to refractive differences that shift apparent distance and angle. In those initial moments, acceleration is not yet at maximum. A leopard seal accelerating from depth can exploit that window, using counter-shaded camouflage to blend into the dim underside of the ice until the final strike. The ecological twist lies in the very strategy that often saves penguins: synchronized mass entry. By entering together, they dilute individual risk through predator confusion and statistical probability. A seal can capture only one penguin at a time. However, this same synchronization creates predictability in timing and location. Predators learn tidal patterns, breeding cycles, and colony routines. The shoreline becomes a scheduled ambush corridor. Energy economics sharpen the stakes. Penguins are endothermic birds with high metabolic demands. especially during breeding season when adults must forage efficiently to sustain both themselves and chicks. Each trip to sea represents a calculated balance between caloric gain and predation risk. Delaying entry too long wastes time and energy. Entering without adequate assessment risks death. The margin for error narrows during periods of food scarcity when longer foraging trips are required. Biomechanics amplify the consequences of blind commitment. Once airborne during the dive, a penguin cannot redirect trajectory. Its flippers are positioned for streamlining, not braking. If a predator positions itself just beneath the expected splash zone, the penguin's descent vector intersects with lethal jaws before evasive maneuvers can begin. Reaction time underwater is rapid once swimming, but zero while falling. Environmental variability complicates assessment. In turbid waters churned by storms, visual detection of predators becomes unreliable. Surface glare under low-angle sunlight can obscure subtle movements below. In such conditions, even vigilant penguins may misinterpret cues. The ocean's optical properties shift hourly, altering the reliability of sensory input. There is also a physiological adjustment during immersion. Peripheral blood vessels constrict as cold water contacts plumage, redistributing heat to core organs. This thermoregulatory reflex, while essential for conserving energy, may momentarily divert neural processing resources. A penguin's body is executing multiple survival programs simultaneously: thermal regulation, buoyancy control, and propulsion initiation. Cognitive bandwidth is finite. Yet penguins are not naive. Many species exhibit probing behavior, leaning over the edge to peer into the water, sometimes entering partially before retreating. Some individuals may serve as inadvertent test divers, their survival informing the group's decision. This appears harsh, but in evolutionary terms it distributes risk across the colony. The death of one may reduce the probability of multiple losses. The narrative reversal emerges when recognizing that hesitation, often interpreted as fear, is adaptive intelligence shaped by predator pressure. Penguins that survived ancestral seas did so by refining their threshold for commitment. However, the cost of false negatives, perceiving no predator when one is present, is fatal. The cost of false positives, delaying entry when no predator exists, is energetic inefficiency. Natural selection tunes this balance, but it cannot eliminate uncertainty. Climate change introduces new distortions. Melting ice alters shoreline geometry, changing traditional entry points and creating unfamiliar bathymetry. Predators may exploit newly formed channels that funnel penguins into narrower corridors. Increased storm frequency can reduce water clarity, amplifying the difficulty of visual assessment. Anthropogenic noise may further mask the acoustic signatures of approaching seals. Once submerged and swimming at full capacity, penguins become formidable. Their wing-propelled locomotion allows tight turns and rapid bursts of speed that rival many fish. Counter-shaded plumage reduces visibility from both above and below. In open water, their agility can outmaneuver predators. The danger concentrates at the threshold, the moment before transformation. A colony gathers at dusk, the horizon painted with fading light. Hunger presses from within. Chicks await regurgitated meals rich in krill and fish. The sea appears calm. One bird steps forward, then another. A collective surge follows. The splash zone erupts, and beneath it a leopard seal angles upward, calculating intercept vectors based on years of learned behavior. The first seconds determine outcome. If the penguins have read the water correctly, they disperse underwater in coordinated arcs, exploiting three-dimensional space to evade pursuit. If they have misjudged, the sea claims one in a burst of crimson diffusion that vanishes into currents. The colony continues. Evolution has normalized this arithmetic. The mistake is not diving. Diving is necessity. The mistake is surrendering to momentum without information. In ecosystems defined by edges, ice to ocean, land to sea, the most dangerous instant is the leap itself. Survival belongs to those who pause long enough to see the shadow beneath the surface before committing to the fall. The water detonates into silver panic. A school of squid erupts from the reef edge in a synchronized blast of jet propulsion, siphons flexing, mantles contracting with muscular violence that turns water into thrust. For a fraction of a second, the maneuver is flawless, hydrodynamic escape driven by rapid mantle cavity compression, expelling water in a focused stream that converts stored elastic energy into acceleration. The squid streak forward like living torpedoes, chromatophores flashing pale in alarm. And then one breaks formation. It jets not into shadow, but into open blue. In that single directional error, speed becomes betrayal. Jet propulsion is a marvel of evolutionary engineering. Unlike fish that rely on lateral undulation, squid generate thrust by drawing water into the mantle cavity and forcefully expelling it through a narrow siphon, achieving bursts of acceleration that can rival many predatory fish in short intervals. Their muscle fibers are tuned for rapid contraction, optimized for escape rather than endurance. Neural circuits within their giant axons transmit signals at extraordinary velocity, coordinating mantle compression with fin stabilization. In controlled circumstances, this system is devastatingly effective. The problem is not speed. The problem is predictability. Predators in pelagic environments do not chase blindly. They calculate vectors. Tuna, billfish, and dolphins possess neural architectures specialized for motion detection and trajectory prediction. Their retinas are packed with cells attuned to contrast and directional change, allowing them to lock onto sudden acceleration in a cluttered visual field. When a squid jets abruptly, it generates turbulence and contrast that ripple outward through water, readable through lateral lines and acute vision. The burst that creates distance also paints a target. In chaotic situations, predators anticipate that prey will flee along the path of least resistance, away from immediate threat and toward perceived open water. That expectation becomes a hunting strategy. The ecological twist lies in the illusion of freedom within speed. A squid fleeing instinctively may believe that maximizing distance equals safety, yet the ocean is a three-dimensional chessboard. Open water offers fewer obstacles, fewer hiding places, and greater visibility. By choosing a direction that prioritizes immediate clearance over structural refuge, the squid trades short-term separation for long-term exposure. Coral overhangs, kelp forests, and thermoclines provide sensory distortion and cover. Open blue amplifies silhouette. Under pressure, the nervous system defaults to reflex pathways, rapid contraction, explosive thrust. But reflex is not strategy, and predators know it. Energy expenditure compounds the risk. Jet propulsion is metabolically expensive. Each burst draws heavily on anaerobic pathways, producing lactate that accumulates in muscle tissue. Unlike fish with red muscle adapted for sustained swimming, squid rely primarily on fast-twitch fibers that fatigue quickly. A high-speed escape can drain energy reserves within seconds, leaving the animal temporarily compromised. Oxygen debt builds as hemocyanin in the blood struggles to deliver sufficient oxygen to tissues. After a frantic jet, the squid may enter a brief period of reduced responsiveness, a recovery window in which reaction time lags and coordination falters. If the initial direction was flawed, there may not be energy for correction. Disorientation adds another invisible cost. Rapid jetting destabilizes spatial awareness. Squid possess complex statocysts, organs analogous to inner ears, that detect orientation and acceleration. In turbulent water, these sensors can be overwhelmed, producing momentary confusion about up, down, and lateral alignment. A predator accustomed to high-speed pursuit maintains visual lock even as prey tumbles. The squid, meanwhile, may overshoot intended cover, emerging into brighter water where its chromatophores struggle to match shifting backgrounds quickly enough. Camouflage requires microsecond neural calibration. Panic scrambles that precision. Consider the dynamics of coordinated hunting. Dolphins hunting squid often operate as pods, herding schools into compact spheres before forcing them upward toward the surface. When the pressure mounts, squid erupt in multiple directions, each individual betting on its own velocity. Dolphins anticipate this fragmentation. They accelerate not toward the densest cluster, but toward the anticipated breakout vectors, intercepting those that choose upward or outward escape paths. In such scenarios, the squid that darts fastest into open water may be the first consumed. The mistake is not weakness, but direction, chosen under neurological duress. Even solitary predators exploit pattern. A tuna tracking a squid will often angle its pursuit slightly ahead of the prey's trajectory, predicting where the jet will carry it rather than where it currently swims. This interception strategy relies on understanding that prey under threat rarely double back immediately. They commit to forward acceleration. A squid that instinctively jets straight away from the perceived source of danger aligns itself with the predator's predictive model. Only by breaking expectation, zigzagging, diving into vertical gradients, or releasing ink to obscure orientation, can it disrupt calculation. Yet ink release itself demands energy and precision, and deploying it too early or too late diminishes its value. The ink cloud represents another layer of complexity. Composed of melanin-rich particles suspended in mucus, the cloud creates both visual obstruction and chemical confusion, potentially interfering with predator olfaction. However, ink is finite. Once expelled, regeneration requires metabolic investment. A squid that wastes ink in a misdirected escape may find itself without this resource during a subsequent encounter. Defense, like speed, is bounded by energy budgets. The narrative reversal deepens when considering that predators, too, risk miscalculation. A squid that darts toward a vertical temperature gradient, a thermocline, enters water layers of differing density and light transmission. Rapid changes in refractive index can distort silhouettes, buying precious milliseconds. In some regions, bioluminescent plankton flash when disturbed, creating bursts of light that may confuse both hunter and hunted. Here, the squid's erratic movement can trigger luminous distraction, turning panic into camouflage. What appears as chaotic flight may, in certain contexts, be a gamble that leverages environmental physics. But such outcomes depend on familiarity. Juvenile squid, inexperienced in the three-dimensional complexity of currents and gradients, are more prone to directional errors. Their neural circuits, though rapid, lack the fine-tuned behavioral repertoire of mature individuals. In schooling species, collective movement can mitigate this vulnerability. Coordinated jetting produces a swarm effect, overwhelming predator focus. Yet cohesion requires split-second alignment. A single individual veering off becomes isolated, its silhouette singular and vulnerable. Predators exploit these fractures in unity. The cost of speed reverberates beyond immediate predation. Repeated high-intensity escapes elevate stress hormones, altering metabolic priorities and potentially impairing growth or reproduction. In ecosystems where predator density is high, squid populations may adapt by modifying average escape responses, favoring shorter, more frequent bursts rather than maximal sprints. This modulation reduces fatigue, but may also reduce immediate separation distance. Evolution balances these trade-offs continuously, sculpting nervous systems and muscle composition in response to predatory intelligence. Ultimately, the wrong direction is not a moral failing, it is a statistical misalignment between reflex and context. The squid's body is engineered for rapid displacement, yet displacement without situational awareness can amplify risk. In the open ocean, survival hinges not merely on speed, but on vector selection. The predator reads patterns, the prey must disrupt them. Under pressure, the brainstem chooses acceleration before the cortex-like structures responsible for complex processing can intervene. Instinct overrides analysis. And still, countless squid survive. Their escapes, when aligned with cover or gradient, are breathtakingly effective. A well-timed jet into kelp shadows or beneath a rocky ledge transforms pursuit into futility. Ink deployed at the moment of directional change can create a decoy silhouette, misleading a predator into striking empty water. In these instances, speed is salvation precisely because it defies expectation. Yet the ocean remembers the failures more efficiently than the successes. A squid that jets into glare, silhouette stark against sunlight filtering from above, becomes an outline visible for meters in every direction. A predator converges, trajectory optimized, muscles coiling for impact. The water churns again, not with escape, but with capture. Energy spent becomes energy transferred up the food chain. In this ecosystem of anticipation and reflex, the difference between life and death may hinge on a few degrees of angle, a slight tilt toward shadow rather than brilliance. Speed is seductive, it feels decisive. But without directional intelligence, it can funnel prey into precisely the corridor predators expect. The mistake is not failure to flee. It is fleeing along the path that hunters have already mapped. The squid's mantle pulses once more in the vast blue, siphon aiming, muscles primed. Around it, invisible vectors intersect. Currents, light gradients, predator trajectories, and neural impulses racing through giant axons. The next burst will either intersect safety or align with prediction. In that silent calculation, the ocean waits, indifferent to panic, attentive only to physics and probability.